to tell you all that the participants here are from school as well as colleges. So I welcome my class 12 students as well as undergraduate and postgraduate first year students. So I hope the students here are present from Noida International University, Shaheen College, Karnataka, Sir Padmavat Singhania University, Rajasthan, JP Institute of Information Technology, and Indraprasth Engineering College, Ansal University, Baba Central University of Punjab, Gautam Buddha University, Maharishi Dayanand University, RD Patil College, Delhi University, Agra College, Sardar Patel University, and in the schools I can see that we have got nominations from Vidya Bal Bhavan, DPS RK Puram, ICSC Srinagar, Kerala Samajan Model School, North Point Children's School, Lucknow Public School, Tagore International School, Vidya Bharati Chinmaya Vidyalaya, Army Public School, Kendriya Vidyalaya, Guru Gobind Singh Public School, Mission Public School, and DAV Public School. So welcome you all. It's great to have you here, and we hope to have a good learning in next 4 weeks in different sessions. I'll start with the introduction of biotechnology in IIM, what we people do and what is our specialization before going ahead with the module. Okay. So is my screen visible now?
Yes, ma'am.
Yes, ma'am.
Okay. So here we have both BTech and BTech and MTech programs in biotechnology. In ILM, we had started the first batch of BTech biotechnology in 2002, and till now 14 batches have passed out. And we have started MTech in biotechnology in 2012. So 5 batches of MTech have been passed out from our college till now. We started biotechnology here with the vision. Just the waiting room has to be taken care of. Roma, please take care of the waiting room. Regularly the signals are popping in from the waiting room. I'm tired. Just a second, students. There seems to be some problem. There seems to be some problem. I'll just get back. 1 second. Okay. So the vision of biotechnology here is to be the center of eminence in the frontier areas of modern biotechnology, and we have been working on making our students world-class technocrats who can identify all the problems of engineering and develop the solutions. Talking about the USP, I want to know whether the students know about the NBA or not. Are you able to unmute yourself? I want to know whether you know the concept of NBA or not. What is NBA?
National Board of Accreditation. Is
Yes.
basically for the courses accreditation.
Yes, exactly. NBA is National Board of Accreditation, and the institutes who have got NBA, it's a benchmark of quality. It's a benchmark of quality education in terms of the lab facilities, in terms of the infrastructure, in terms of the faculties, in terms of the teaching methodology. So I am glad to share that Department of Biotechnology at ILM has got accreditation from NBA thrice. The first accreditation was obtained in 2009. Then we again got in 2013, and latest we have got in 2018. In biotechnology, we have got 7 labs here, and these 7 labs are from fermentation technology. We'll be covering one by one. I like to show you my labs, what all labs we have got here. As I said, we have got 7 labs. So one lab is fermentation technology, whereby we do all the production experiments. We produce ethanol, we produce enzymes in this lab. You can see it's a modern day automated fermenter. The student is working on it. So in my session of industrial biotechnology, I'll be taking you through these module, which will discuss the working of fermentation. Second lab, we have biochemistry. In biochemistry, we do all the colorimetric estimation, protein estimation. The basic biochemistry experiments are done in this lab. Third lab we have is plant tissue culture lab, which is taken care by Dr. Abhinav sir, who will be taking plant technology module with you all. In this, we are trying to grow the callus and make new plants. The most sophisticated lab here is our project lab. This lab is dedicated to MTech students. Whatever high-end project work we do, we use this lab. Then we have got immunology lab. In immunology lab, we do the experiments for estimating the antibodies, for blood typing, all the kind of immunology experiments, be it ELISA, be it radial immunoassay, be it Australoni double diffusion. All these techniques we conduct in our immunology lab. Then we have got genetic engineering lab. In genetic engineering, as you must be knowing that we work on making transgenics. We work on making competent cells. So all these experiments of molecular biology and creating competent cells and making transgenics, these are done in genetic engineering lab. And the buzzword nowadays is bioinformatics. So we have a high-end bioinformatics lab with sophisticated configuration and good softwares for drug designing and modeling. So, and this webinar, the students who have attended one webinar on 8th of May, there also Dr. Roma told you about bioinformatics, what all we do here in this lab. So talking about the first USP, I told you it's an India accredited department who has been accredited thrice. Then we have 7 state-of-the-art lab facilities, and main strength of biotechnology at ILM is its faculty. We have a department with 9 people, and 80% of the faculty members of biotechnology are having PhD. We have different expertise. If I talk about myself, I am Dr. Pallavi Singh. My expertise area is microbiology and genomic toxicology. Your second module will be taken by Dr. Roma Chandra on bioinformatics. She has a specialization in bioinformatics and biochemistry. She has done her MTech from IIIT Allahabad and is having PhD from Sir Venkateshwara University. Your third module on plant biotechnology will be taken by our esteemed scientist in the department, Dr. Abhinav Kumar. He is having a lot of projects. He's associate professor with expertise in plant biotechnology and virology. Sir has got sponsored research projects from SERB and AKTU, many funding agencies, many students at ILM in BTech and MTech are working under his guidance and have published good research papers of repute. And our fourth member, who's a very strong pillar in biotechnology, is Dr. Nitin Garg. He is associate professor and is having expertise in plant biotechnology, molecular biology, and IPR. Nitin sir is having a vast experience from last 17 years, and I am very much sure you people will be benefited from his expertise in IPR. He will be taking you through this module. Talking about the next USP, as I said that department is made by its faculty as well as its students. So our students are a matter of pride for us. We have the students who have got funded research projects from UPCST. UPCST is UP Council of Science and Technology. So our undergraduate students in BTech have got the projects funded from UPCST. And I will show you the certificates. These are the certificates of my students from UPCST, which showing that they have got the funding and their projects are up to the mark of national competence. And another matter of pride for me is that every year in AKTU, the Department of Biotechnology gets its students in top 20. We have our students in top 20 every year, and they have been continuously cracking the examinations like GATE. Apart from that, in 2018, our students won the international challenge in USA in an innovation challenge at PRME. PRME is Principles of Responsible Management in Education. It's a breakthrough innovation challenge which was given to the students, and they developed a model on precision agriculture. And they obtained first prize at Asia level in that competition. So I'll show my students. This is one of my students, Ruhi Rastagi. She got 6th position in 2016. Then we have many others who have a scored 10th position, 11th position. I have put photographs of my some students who have cracked grade in 2017. Two of them had qualified gate in 2015. 4 gate qualifiers were there. Then this is the team. The guide is there, Dr. Neha Shrivastava, in the department along with the students. They are at United Nations PRME, and this is the team which had won first prize at Asia level by making a model on precision agriculture. Another very important aspect of our department is that to boost up the industrial skills of the students and give them real exposure, we organize the industrial visits to reputed companies like Yakult, Parle-G, Anmol Bakers, NDRI, Coca-Cola to accentuate opportunity to be in top-notch research and academic organizations. We provide them summer internship. Are you able to hear me?
Yes.
Yes, ma'am.
Yes, we
Yes,
give them
ma'am.
summer
Yes,
internship
ma'am.
every year. We are sending our students for summer internship in good research and academic organizations like CSIR Labs, CDRI, IGIB, AIMS, Himalaya Drug Company, NICPR, IIT Delhi, IRI. This is a good measure to boost up their research skills as well as their aptitude. Then why we are able to get our students crack the national examinations, because in the curriculum we have incorporated the classes for the competitive examinations of GATE and GRE. So every week the faculty members take 2 to 3 classes for these examinations. Then apart from the academics, we also take care of extracurricular activities. We have a departmental club, iBioc. iBioc means I am the biotechnology club. I have to disable the annotation. Okay. So these are the regular activities which are conducted by the iBioc: debate, poetry, painting and drawing competitions, science quizzes, paper competitions, singing. All these we do round the year. And any institute cannot flourish without good placements. So our students are getting placed with good package of 7 to 8 lakhs per year. The maximum package this year was 12.5 lakhs. So these are the companies: Mankind Pharma, Coca-Cola, Nestle, Jubilant, Penicia Biotech, Lifecell, Cryoviva, Aptera, Max, Kingfisher, Wipro, Ranbaxy, Biocon, Codlife, which are the top recruiters of BTech biotechnology students here. We also believe in organizing the conferences, workshops, and guest lectures around the year. So Biogenesis is our landmark, is our main conference that we organize every year. The different series have been organized of Biogenesis. Till now, we have organized 6 conferences under Biogenesis. So here we organize, we invite national and international experts from various organizations to give insights on latest developments in fields of biotechnology. So I have shared the photographs. We also keep on giving the guest lectures. Like this is one of the pictures of guest lecture of Professor Veena Kaul, head of Center of Biomedical Engineering at IIT Delhi. She gave a good lecture on insights of biomedical nanotechnology. This is a glimpse of the healthcare session conducted by Yakult. Then we had invited Professor T.P. Singh from AIMS. He gave a very good insight on development of antibiotics and how can we use molds to make antibiotics and fight against invading microbes. Then we believe that if our alumni are happy with us, if they are handholding the current students, the students will be benefited. So in order to give a brief of what went on in industry and what is the actual situation of biotechnology, we keep on inviting our alumni. Like this is one of our alumni, Mr. Agam Chaudhary of second batch of ILM, 2007 pass out. He is currently the managing director in iDream Digital Labs. He came and gave a session on career of biotechnologist in digital marketing. Then we also organize the workshops, hands-on workshops on IPR, on pharmacovigilance in clinical trials, on natural product research, a lot of workshops. Every year on an average, we organize 3 to 4 workshops for our students. And as I told you about the industrial visits, so these are the places: Anmol Baker's, Mother Dairy, NDRI. I will show you the photographs where the students have gone. This is a picture of Moon Beverages, a unit of Coca-Cola, then Mother Dairy, and NSIC, all these places. Every year we are taking them to the to one or two industrial visits. So I hope you have got a good flavor of biotech department at ILM, what we do, what is the faculty, what is the expertise, what are the activities. Now I'll come on to the modules. So in this training that you have enrolled, we have planned 4 modules. Our first module is Introduction to Industrial Biotechnology, which will be taken by me in this week. I'll be covering how to design the media, what are the various types of the culture, batch culture, fed batch culture, continuous culture, what is the kinetics of sterilization, how do we optimize the industrial processes for making good products, and what are the various challenges in the domain of industrial biotechnology. And along with me, Dr. Roma will be covering the basics of biochemistry, like biomolecules and their structure, to give you a glimpse of biochemistry. And second week, we will be covering Introduction to Bioinformatics, taken by Dr. Roma, myself, and Abhinav sir. Third week is dedicated to plant technology, which will be taken by Dr. Abhinav and Dr. Nitin. And in week 4, we'll be talking about IPR and recent techniques in biotechnology. The module leader for IPR is Dr. Nitin Garg. Techniques will be taken by all of us. So from this week to week 4, you will have an idea about these techniques, and we will also give you the virtual tours, virtual lab sessions, and there will be the expert sessions from the industry. As I had shared with you in the email also, that these are the experts: Dr. Abdul Aziz from Michigan Technical University, USA, Dr. Rakeshwar Bandichor from Dr. Reddy's, Professor M.V. Rajim from University of Delhi, Dr. Hriday Ranjan from AIMS, Dr. Khalid Raja from Jamia Millia Islamia, Dr. Madhu Chopra from DU, Ms. Pooja Gupta from USA, Preeti Tyagi, Mr. Shivdat Sharma from Nestle, Mr. Ankur Chaturvedi from Genes2Me Diagnostic Labs. So I had shared with you yesterday the session, at what time, at what date, on what topic they are going to take the lectures. So this schedule which I have put here, today we are having 2 inaugural lectures. Ms. Pooja Gupta will talk about CAR T-cell therapy, and Professor Rajam will give you details about plant technology. What is the past, present, and future directions? So Professor Rajam is a very renowned professor in University of Delhi, and he has served for more than 30 years. You will be gaining a lot from his experience. That's all from my side. Now I invite my fellow colleague, Dr. Abhinav, to give you a layout, to give you what he is going to cover in his module on plant technology.
Good morning, all. Good morning, all the participants.
Good morning, sir.
Good morning, sir.
Good morning,
morning,
sir.
Sir.
Good morning, sir.
Good morning.
Good morning, sir.
Thank you, Dr. Pallavi. You have set the tone for today's start the inaugural session for our 4-week Bioescalator program. I will be taking care of plant biotechnology and plant sciences. The topics which I will cover ranges from cell therapy, cell theory, sample of growth like plant regeneration, plant tissue culture, culture media, media components, aseptic techniques, and definitely there will be some virtual labs from which you will take benefits. Plant tissue culture technique is not a new technique. A lot of plants have been regenerated across the globe with the help of this technique. And I must say, this is one of the best idea to start a startup in our country, because of very few companies, very few initiatives are taking care of this domain. So it will be a good for your career also. Power of regeneration is very high for a plant. A plant, if I say, is a virtually immortal, then I am not wrong. And in the last, I say that plant tissue culture lab, the idea covers the media preparation room, culture media, how you wash, every details technique will be covered in my module, aseptic transfer chamber area, environmentally controlled culture rooms, analytical rooms, acclimatization, how you acclimatize it, and several miscellaneous items like air conditioner, marker, matchbox, burner. So at the end, I just want to say that this plant science and plant biotechnology, I used to call it BMW. And you must know this word is BMW. At the end of the lecture, at the end of my module, you come to know that why exactly I am telling this plant sciences and plant biotechnology as BMW. So thank you, Dr. Pallavi. So the next session will be, next module, a description will be given by Dr. Roma Chandra. She has a vast experience of bioinformatics of 10 years. So Dr. Roma Chandra.
Yes, thank you so much, sir. I'll be starting with bioinformatics, and you must be knowing what is bioinformatics. So, bioinformatics is actually the study of biological information using computational techniques. So, in my module, I'll be covering all the basics of bioinformatics. So, we'll be starting with the biological databases, then we'll proceed with the bioinformatics tools, and then I'll be covering the various applications of bioinformatics. So I'll start with the sequence alignment process, then I'll continue with the process of protein structure prediction, how we can go with the functional analysis, what about the visualization, what about the various softwares which we can use for all these purposes. Then we'll go with the gene prediction process as well, how we can use the bioinformatics tools to study the whole genome. Okay, and then later on we'll just go with the study of phylogenetic tree, the evolutionary tree study, and the last module, the part of the module will be the drug designing. So we'll talk about computer-aided drug designing. So I'll cover all the things which will be under this CAD, including the structure-based, ligand-based drug designing, the docking process, the QSAR process, and so on. So Dr. Abhinav and Dr. Pallavi, they both will be there in my module, and we'll just cover the practicals as well during this bioinformatics sessions. Okay.
Thank you, Roma. I now request Dr. Nitin Garg to provide what he is going to cover in his module on IPR.
Good morning, everybody. Myself is Dr. Nitin Garg, and in fourth module of IPR, I will cover about various aspects of IPR. What basically IPR is, how we can use the IPR in our research. As all the fields of biotechnology, or you can say that all the fields of science and technology require certain type of protection, and that protection you will get from IPR. In my modules of IPR, we can study about what actually IPR is, what are the various forms of IPR, such as patent, copyright, trademarks, barcode, trade secret, industrial designs, geographical indicators, how you can protect your research from IPR. What is the basic difference between discovery and invention? We can also study the three important characteristics of IPR, that is distinctiveness, uniformity, and stability. In our module, we can also study about GATE, General Agreement of Trade and Tariff, TRIPS, trade-related intellectual property aspects. We can also study about transboundary movement in the field of IPR. How one country can get genetically modified organism from another country. How one country can utilize the genetic resources of other country. How the benefit gets shared. In this, we can also study role of IPR for biotechnology. How a biotechnology student can get benefit from the IPR. There are various companies in India, various universities can hire patent attorneys. How you can become a patent attorney. In our module, we can also study about how to file a patent. What are the procedure to filing a patent? What are the steps to file a patent? So all those things we can cover in this particular module. Thank you.
Thank you, Nitin sir. I now request Dr. Roma to introduce our speaker today, Ms. Pooja Gupta. Welcome, Pooja. We are very happy to have you here. Are you able to hear me, Pooja? Roma, please introduce her.
Yes.
Hi, so much.
Okay, so I'll introduce Ms. Pooja Gupta. She is actually presently working as the lead quality analyst in the quality control department of Bristol Myers Squibb in Seattle, USA. And there she is handling the planning and scheduling division. And prior to this, she was actually working with the LabCorp that is in USA and Ondyne Incorporation. She is actually from India itself, and she had completed her MTech from IIIT Allahabad. She had qualified GATE, and she is having an experience of industry and academic for about 11 years. She is having various publications in national and international journals, and she is actually a lover of science and trekking. So I'll just request Pooja ma'am to please start your lecture.
Hi everyone, and thank you so much Roma ma'am for having me here, and Pallavi ma'am and everyone else. Nitin sir, hi. Okay, so let me start, and I'm not able to, it's disabled, so can you please enable?
Okay, just wait.
We need to make her co-host.
Yes. Just wait a second. Pallavi ma'am, you made her?
I'm going to make her, just a second. Pooja, now you can share.
Yes,
I
thank
you.
Ma'am, can you please, ma'am?
Yes.
Ma'am, can you please allow to record the Zoom meeting for today's class?
We are already recording. You will get a recording of this on the YouTube channel of the college. Okay?
Okay. Okay.
Hi, so can everyone see my screen?
Yes, Pooja, yes.
Okay, perfect.
Yes, ma'am.
So let's start with CAR T-cell therapy. So if we talk about CAR T-cell therapy, it's a wonderful and it's a exciting new type of therapy for cancer. My name is Pooja Gupta, and I work with Bristol Myers Squibb in Seattle, Washington. And let's start with the therapy and what it says. So if we talk about immune system, our immune system consists of mainly two type of immune response. One is if we talk innate immunity, and another is adaptive immunity. And what does that mean is innate immunity, if we talk about, it's very basic and it's the first line of defense. And the adaptive immunity is the T cells and B cells, and that's how it become interesting. Why? Because T cell and B cells secrete antibodies, and these antibodies basically combine with the antigen. Now, what is antibody and what is antigen? So basically, if we talk about the antibodies, so T cells and B cells secretes antibodies, and our target cell contains the antigen on its surface. So this is if you see the slide, you can see here it is a diagram of T cells. So it's a very basic, very basic diagram, and it shows the TCR, which is T cell receptor. So T cell receptor consists of two main parts. One is extracellular part, which is this outside of the cell, and one is intracellular part. So whenever there is an antigen which is present on the target cell, say for example, here I'm talking about cancer. For example, a cancer cell is having an antigen on its surface. So T cells, TCR, combine or attached with the antigen, and then it provides the signaling to the intracellular ignition switch here, and it turns it on, and it kills the cell. That's how it happens. So usually and I hope none of us is having any cancer. And why? Because our T cell is working fine, and and thanks to the, you know, any target cell which represents its antigen via MHC. So this portion, the block on red, is MHC, major histocompatibility complex. So if an antigen is present on the MHC, then only T cell can identify and attach to it and basically kills it. So here is the diagram which just shows that antigen, which is in bright red color, is presented to TCRs via MHC, and then that's how the T cell get activated and basically it kills the cancer. So can it happen that a cell is having antigen, but it's not presented via MHC? Yes, it can happen. And this is basically a escape mechanism for cancer cells. That's how they escape from getting killed by T cells. So here you can see there is antigen present on the surface, but there is no MHC. So, T cell is kind of a prince. He don't eat its food until it's served on silver platter. So here, because MHC is not there, it's not going to kill it. So that's what it's shown there. So now the issue with that issue is that T cell need to have MHC on in order to attack a target cell. But if there is no MHC, then this thing cannot work. So we need a receptor which can actually work even if there is no MHC on the target cell. So that's how the chimera or chimeric word came from, because scientists are brainstorming and they came up with an idea. So it's a Greek mythology which says if you combine like different animals, or you combine different natural things and take advantage of different things and make another thing which is more stronger. So that's what they are doing. So next thing is like chimeric antigen receptor, which we call CAR. So we all know that antibodies can bind with the antigen, and then there is no need of MHC. Antibodies can directly bind to the antigen without any need of MHC. So what they have suggested, or they thought, is that if we can just take this antibody structure, like the outer structure of antibody and the inner structure of TCR receptor, and then combine these two, and then they can make a more stronger type of receptor, which we call CAR receptor or chimeric antigen receptor. So that's what they thought about it. So, and another thing they thought is that instead of just replacing this TCR with CAR, if they can just attach one CAR on the surface of T cell. So what's the benefit of this thing? Its benefit is that, for example, there is a cancer cell which has antigen on its surface, and antigen is presented via MHC, then this regular TCR can kill it. Another scenario, if a cancer cell is there which has antigen but which don't have any MHC, then this CAR, which they artificially synthesized, can bind, because I have shown you here, it has ability to just bind to any antigen. I mean, not any antigen specific to the antibody, but still it can bind if even if there is no MHC molecule. Okay, here. So this is basically a CAR T cell. And now the question arises how they insert this. So basically, this is a protein, like CAR is a protein, and how a protein is on the cell. So protein need to be genetically engineered, and need to be genetically engineered in the same way that they can secrete their own CAR on the surface of T cell. So how this transfection can take place, how external DNA can be in a cell. So there are a few methods like lentivirus can help, retrovirus can help, electroporation can help, or basically there are so many genetic engineering methods which can be used to insert the CAR gene in the T cells. So that's how one of the methods, I mean, in my company, lentivirus is used. So we use lentivirus to infect the T cells, and this happens in the lab. Okay, so that's how we introduce CAR in the T cell. So now there is one more thing apart from that. So this green structure, which is half inside and half outside, is our TCR, T cell receptor, and this is natural thing, you know. So nature already have its own co-stimulatory domains, which is not shown here in the figure. But this CAR, the yellow portion, the CAR is synthetically made. It is artificial. And scientists noticed that though this CAR attached to an antigen, but this don't activate to a larger extent the T cell. So what they need some help. The CAR needs some help. So they synthetically introduce co-stimulatory domains, which are shown here in purple. So basically this TCR has its own co-stimulatory domain, but the CAR don't have. So they artificially introduce another thing. So if you can see, CAR is a pretty complex structure. It has its antibody binding site, antigen binding site, which is a portion of antibody, and then it has its transmembrane part and then intracellular part, and along with that it has its co-stimulatory domain. So what this help is in activation of this T cell. So whenever that T cell get activated, and how it get activated, whenever there is a antigen which binds either here or here, it activates the T cell. And what happens when the T cell activate? It divides, because it's a cell, so it divides whenever it activates. And how does it activate? So activation here means antigen is present. And antigen is present means a cell is present, or a cancer cell is present, which has antigen on its surface. So that's how it get activated. And whenever it get activated, the parent cell divide into two daughter cell, and then again daughter cell divide into two more, and so on and so forth, like 2 raised to the power, and you know that how it divides. And that's how it multiply its army. So for example, I'm talking about T cells. So T cells are basically in your body, correct? So what scientists and what like companies do is they take out the blood from your body, just like you donate blood, right? So you go to hospital and then they make you sit, and the same way that you donate blood, they basically extract T cells from your regular cells, like regular blood cells. And this process is called apheresis. So leukopheresis and or apheresis, it's same thing. It's just white blood cells, so we call it leukopheresis. Okay, so the procedure of basically separating white blood cell, that is B cells and T cells, from the RBC and plasma and whatnot, is called apheresis. And with that procedure, we have T cells and B cells separated from your regular blood, and then they ship them to a manufacturing plant, or maybe if it's something is going on in academia, they just ship it, and there they transfect it with some virus so that the CAR gene get inserted. And after the CAR gene is inserted, these cells are harvested again. Like you have to increase the number of cells, so it need to be harvested. You grow them basically, and then you harvest them, and then these modified cells are basically shipped back to the hospital where patient is actually waiting. I mean, not exactly waiting, not sitting there, but it's the whole thing is it takes like manufacturing of CAR T cells takes about 9 to 10 days. So after 9 to 10 days, when this cell is manufactured, then it shipped back to a hospital, the same hospital where your patient is, and that patient basically receives this T cells which are modified. And if there is obviously the therapy is only going to take place, or your doctor is only going to allow for this therapy if you have consistent cancer and if there is no other treatment is working for you. So if when we administer the T cells, the modified T cells, then these CAR and TCR attached to the antigen which is present on the cancer cell, and that's how it get activated. So basically, if there is cancer cell in your body, then only T cell get activated. Otherwise, it won't get activated. And another thing is, so for example, imagine like doctor inserted like with the help of IV. Basically, it's just a sort of injection. So basically, T cells get administered in a patient body. For example, he administered like 100 CAR T cells. Then via this procedure, what we are seeing here, they get multiplied. Can you see, like these all are multiplying? And why they are multiplying? Because your body is having cancer cell, cancer antigen, and that's how it get activated, and that's how it's multiplying. So that's how the concept of living drug came up. So basically, it's a drug, and why we are calling it a drug? Because it's killing the cancer, right? So it is a type of drug, but it's a very different kind of drug. Why? Because it's not a vaccine. It's not it's a living cell. It's not a vaccine. It's not an antibody. It's nothing like that. But it's a living cell, and this drug is basically dividing in your body. So unlike other medications, it's not going to deplete after 1 hour, 2 hour, or 3 hour. For example, you have like fever, so you take paracetamol, and paracetamol works on the cells or the target, and then after like 4, 5, 6 hours, you again have to take another paracetamol tablet. Why? Because it's degrading in your body. And here its opposite thing is going on. If there is cancer cells still, it's gonna multiply. So the cells are basically multiplying. If and it's not going to just multiply like randomly. It's only going to multiply if there is antigen. So if there is no tumor or all tumor is killed, then it's not gonna multiply. So that's the best part. It's a living drug. It's just one time thing. You need not to go through again and again. So that's the main part of this. So if we talk about how it works and how it looks like, it's looked like something like this. So CAR structure is given on the left side of the slide. So you can see the antigen binding site is present. So this portion, antigen binding site, basically is part of antibody, and then there is transmembrane domain, which is between the membrane, and then internal intracellular membrane is intracellular domain is there, which do signal transduction. So here we can see the mechanism. So the antigen binds with the CAR, and it gives signaling to the inside of the cell, and that's how it get activated. And when it get activated, it basically call other its friends like other B cells, plasma cells, dendrite, or maybe cytokines, and then some other enzymes, and then they all together work on that cancer, and that's how it get killed. So basically, it's a response from all the immune cells, but basically we can say T cell is the main killer cell because it has the ability to bind with the antigen even if there is no MHC. So CAR basically help to bind with the antigen even if there is no MHC. So the regular TCR, of course, is not shown here, but this is also present here, and that can also bind to a tumor cell, but only if MHC is present. So that's the difference between CAR and the regular TCR. And obviously, if regular thing is working, if your regular TCR is killing the tumor, then you need not to go through this therapy. So mainly we are going through the therapy because your regular thing is not going to take place or not working well. So now there are various generations of CARs. So if you can see, the difference is only about how it provides signal to the intracellular portion of the cell. For example, there can be like, so main thing is if we go back, okay, sorry. Yeah. If we go back here, so main thing is like signaling. So how strong your signal is and how accurate your signal is and how fast your signal is. That's how it's gonna act. This whole immune response is going to take place, like mast cell or dendrite cell and so many other cells that are going to take place only depending on how signaling works. So basically it's showing here that there are different type of co-stimulatory, co-stimulatory, what do you call it? Okay, admit. Sorry, yeah. Different type of co-stimulatory domains. Okay, next. So I have a video which I'm showing you which describe the whole procedure. Let me know if you can hear the volume and everything.
At LLS, we always say that beating cancer is in our blood.
Okay. Okay, perfect.
At LLS, we always say that beating cancer is in our blood. It's a phrase that captures the heart and soul of what we do. But what if your own blood could actually be turned into a meaner, leaner, cancer-beating machine? Well, that's what doctors and researchers are doing right now with something called Chimeric Antigen Receptor T-cell, or CAR T-cell therapy. To understand it, we need to start with T cells, a type of white blood cell that patrols our bodies for signs of disease. See, all cells have protein fragments called antigens on their surface. Some antigens are tumor-specific, meaning they're specific to certain cancers. And it is these tumor-specific antigens that ultimately alert your body's immune system, specifically your T cells. See, T cells have receptors on their surface that are uniquely designed to latch onto antigens of cancer cells and kill the harmful cells by injecting them with toxins. It's a pretty amazing system when it works. The trouble is, cancer is smart. Cancer cells can disguise themselves so our T cells don't recognize them as foreign. And when that happens, the cancer grows, leaving one of your body's most powerful defenses, well, kind of powerless. So researchers asked themselves, what if we could reprogram a person's own T cells to break through cancer's defenses? And that's what CAR T-cell therapy is all about. In a nutshell, the doctor takes out some of your own T cells and sends them to a lab. The laboratory technicians turns them into super cells, CAR T cells. They do this by modifying the new CAR T cell receptors to recognize your specific cancer and make those receptors, well, more receptive to its antigens. New cells are reproduced a few million times and then frozen so doctors can begin treatment when the time is best for you. Before you're able to get your new CAR T cells, you'll be given something called lymphodepleting chemotherapy, which will make room for your new super cells. Then, when it's time, your doctor will introduce the new and improved T cells back into your body. The good news is that they start attacking the harmful cells right away. The bad news is that the therapy has a high likelihood of causing side effects and making you sick. Many patients experience symptoms typical to a bad case of the flu. Patients also report nausea, dizziness, and loss of balance, among other symptoms. And make no mistake, in some rare cases, patients can experience more serious side effects, like confusion, seizures, and even stroke-like symptoms. Your doctors will want to monitor your condition, so they'll likely require you to stay near a hospital or perhaps even check into one. But for many patients, these side effects are temporary and can be managed with medications or other procedures. What's more, researchers are making great strides in controlling and reducing those side effects. So what happens after the sickness passes? Well, this is the really good part. These stronger and faster CAR T cells just keep reproducing in your body for a long time after the infusion, continuing to fight the cancer if it returns. And in many cases, they continue to work long after a patient enters remission. Right now, this therapy is typically only given to patients with specific types of blood cancer who have not responded to common cancer treatments. And only your doctor can tell you if this therapy is a real option for you. But while the results are early, they're incredibly promising. And encouraging studies for other types of blood cancers are moving forward at great speed. As beating cancer is in our blood, but the key to beating your cancer might just be in yours.
Okay, so basically these are the three companies' name. If you just saw that, Bristol Myers Squibb, Novartis, and Kite are the three companies which works with the CAR T cell production. So that's why it's showing. Okay, next. So currently, if we talk about CAR T cell therapy, is it FDA approved? Yes, it is FDA approved. And it's approved for leukemia and lymphoma. Leukemia is, you all know that it's a cancer for white blood cell, and lymphoma is cancer for lymph nodes. There are few more clinical trials are going on, and hopefully very soon some other CAR T therapy drugs will be in market, which will be for other type of cancer, and different companies are still working on this. For example, my company, Bristol Myers Squibb, is working on Breyanzi, which is a T-cell therapy for leukemia. And every company works with a little bit different enzymes and, like transfection method, and like different, I would call CAR. So that's how different companies has a different drug, like CAR drug. Okay, so another portion is how it works. So most of the part is already covered in the video that I just showed you. So what it says that how it works is basically the patient blood. Of course, this all will only happen if your doctor allows for, for example, a patient is having cancer. So what you do, you just go to hospital and then talk to your doctor, and then your doctor do biopsy and whatnot, and then they let you know that, oh, you have cancer or you have leukemia, for example. Then what's the option, you ask, and then they say, okay, there are few methods. What are the methods? Like, what are the treatments? So treatment is like maybe bone marrow transplant or maybe chemotherapy. You might have heard few of them, right? For example, you went through them, like, for example, you went through like chemotherapy, but cancer relapse, cancer, they came back. So it happens. It happened quite often that they just came back after a few years. So if two plan of action failed, then your doctor is going to suggest CAR T therapy. And why? Because CAR T therapy is pretty new, and a few of the things are still in the clinical trial mode. So it's few things are still not approved, like the few are still not approved by FDA. So that's how it happened. Like if there are other therapies, then doctors are gonna suggest you that therapy. But when those therapies are failing on you, then they're gonna suggest you CAR T therapy because it's the ultimate one. Okay, so you go to a hospital, and then here it says the patient basically kind of donate their blood, and then they extract the T cells from that blood. They ship it to a manufacturing plant. Where my office, for example, I work in a manufacturing basically lab. So they ship there, they genetically in the lab, they genetically alter it, and the CAR is introduced, and then they harvest it in the lab, and then they check, the quality check is done to identify that everything is, you know, safe, and whatever is supposed to do is the cells are doing that thing, and then we ship them back to the patient, basically to the hospital, and then the doctor is gonna, you know, administer that dose, and that dose and everything is told by the lab. Whatever, whoever is designing this CAR is gonna tell doctor that whatever is dose will be. Because why? And that thing is very important because it has some side effects. So if the dosage is not correct, it's gonna have some side effect. So side effect part is doctor going to take place, like going to manage with medication and whatnot, but it does happen. So whatever testing we do here, basically in that testing we find out that what dose should be. Okay, so next. Why it's not clicking? Okay. Is my computer hang? Can you hear me?
We can hear you, but the screen is not actually, the slide is not changing. You can just escape the
No, my screen
page.
button is not working. Let me.
So you can just stop your screen share and share your PPT once again.
Okay. Can you see my screen now?
Yes.
Okay, perfect. So here. So I have attached one. Yeah. Okay. So here I have attached one picture which shows the same step, the one which we do in clinic, then in manufacturing lab, then back in clinic, and then what happened exactly in your body, which I have shown you in, you know, initial slides, that how that CAR is going to kill the cancer. So basically, the lab part, the second number, the manufacturing facility is my office where we do all of these stuff, and our clinic is basically Fred Hutchinson Cancer Research Institute. You might have heard of that name. Okay, so here is the CAR T cell treatment schema where doctor screens the patient, because sometimes it's clinical trials. So not every patient or not everyone can participate in a clinical trial, but if you qualify, for example, and what are the qualifying criteria? Generally, it's just if other plan of action or plan of treatment failed on you, then you may qualify for the clinical trials. And then leukapheresis, if your doctor approves, then leukapheresis takes place in the hospital. And then here it says bridging. So basically, bridging is the time when after leukapheresis, T cells are sent to the manufacturing lab, and then meanwhile, doctor will give you some medications so that your body get prepared for CAR T cell therapy. And before the CAR T cell infusion, conditioning chemotherapy is done. What is conditioning chemotherapy? It's a kind of chemotherapy, but very low level chemotherapy. It's not that, you know, so usually chemotherapy, you already know probably that chemotherapy is basically a doctor gives some drugs. You might have, you know, seen few cancer patient whose hair shed and, you know, whatnot. So it's very rigorous mode of treatment. So it's so here with the CAR, it's not that rigorous. It's not like that dangerous, I would call. It's very mild. But why we do the conditioning chemotherapy? Because we still want some, you know, space in your blood for our modified T cell. If the number of T cell is present in your cell and these T cells are not, you know, doing their stuff, so we don't want them. I mean, we want them for few regular T cells are fine, but we want modified one to go in your bloodstream. So we need some space for that modified one. So that's why we do chemotherapy, so that few regular cell dies, so that there is more space for, you know, modified one. So, and this chemotherapy, basically we do like -5. Why we call it -5? Because the day when we infuse the CAR T in the patient, we call it day 0. So like 5 days before that, we do chemotherapy. And after 5 days, we insert via IV the CAR T cell. And then we close monitor. And here, like after 3 or 4 days, what we call is like, there are some side effects. And what are the side effects? Side effects are basically, we call it a CRS, cytokine response syndrome. So basically it's a flu-like symptoms. That's what the video was also saying. It's flu-like symptom, but it's like very bad flu, like the one you never had before, kind of. You know, but it's not a bad thing, you know, and it's kind of good thing and certainly a normal thing. And why? Because it means that CAR T therapy is working on you, you know, because why there is a fever and why there is flu-like symptoms, it means because T cell is killing the cancer cell. So whenever a machinery, you know, run, you might have noticed that when a machinery run for a longer time, if you touch the outer surface, it become warm or hot because something is going inside, right? So same way, like your body is fighting with the cancer. So that's why a fever is there. That's why, you know, flu-like symptom is there. That's why coughing is there. That's why cold is there. And these all things are pretty bad. Like you will feel like, oh, I'm not, you know, I'm getting sick. You feel like you are getting sick, but in reality, you are getting better. So after like 7 days, you'll feel like much better position. And after like 30 days, like around a month, your cancer will be gone. Here, like on day 30, we generally assess the tumor and like it generally just goes away. So that's how it work. But this period, like around seventh day and like, you know, 3rd to 7th day is like pretty bad. You feel like you have, you know, it's dizziness is there and maybe confusion is there. You might faint. You might go to indeed there might be a need to go to ICU, but eventually you will be fine. So it's not a bad thing, I would say. And so why we need CAR T therapy? Because you might have heard that, oh, there is chemotherapy, there is some other type of therapy. Why? Because the success rate is pretty high. Like for leukemia, it's 94%, and for lymphoma, it's more than 80%. It's pretty good. Like none of the other treatment that we have has that much success rate. For example, we might.
Ma'am, it paused.
Sorry. Hello.
Anyone wants to ask something? Ma'am, you can continue.
Okay. So one more thing, you can actually, you know, interrupt me in between, and if you have question, I can, you know, I would love to answer them.
Mummy, वो नमक डाल नहीं डाली
So
हो?
I guess so. There is some kind of issue with the.
Fine. Okay, so you are muted. I'm muted. Why I'm muted? You are unmuted. Okay, I'm unmuted now. Okay, so so there are different type of, you know, cancer treatment options. Like for example, hormone therapy is there. And what is hormone therapy? Basically, every cell needs some hormone, you know, for their flourishment. So hormone therapy is where you just restrict the hormone that required, like the food is restricted, so cells gonna die, that sort of thing. Another thing is like surgery. And what's the drawback of surgery is surgery works on the tumor cell, but if you remove a tumor, some cells might still not removed, and then, you know, cancer relapse. another is bone marrow transplants. So this is the most successful till now, like apart from CAR T therapy. Bone marrow transplant is like changing the machinery, because in bone marrow the T cells and B cells are produced. And then chemotherapy, and chemotherapy is like, one of my friend, she got cancer, and then when she was back in India, she was like 14, 15 years, and she got cancer, and then she went through chemo there, and then she came here on site, like here in US, and her cancer relapsed, and again she went through like chemotherapy, and chemotherapy failed, and then she went for bone marrow transplant, and luckily she's doing fine. But if not, then CAR T therapy would be her another option. When she was going through bone marrow transplantation, that time doctor did talked about like CAR T therapies, that if that thing gonna fail. So there are a few like targeted therapy and like radiation therapy, but all these have some side effects. Like I'm saying, even the immunotherapy, like CAR T therapy, is still gonna have some side effects, but it's not. I would not call it side effect. I mean, it just means that therapy is working, and you have to just deal with it, I would say. Okay, so who we are Bristol Myers Squibb, and so basically I work with therapeutics. Yes. Hello. Any question?
Ma'am, can you please explain why we need CAR T cell therapy again?
Oh, okay. So CAR T therapy is needed because, so generally what happens, like, for example, a patient is there who has, who is like perfectly fine. For example, I'm taking example of you. You are perfectly fine. And why you are perfectly fine? Because even if there is a target cell, even if there is a virus or something, then your T cells are killing that virus or whatever it is, okay? So that's how you are healthy, because your T cells are working just fine to killing that that target cell or that virus, that bacteria, whatever it is. But imagine a situation that someone is having cancer, and that cancer. So basically, how cancer develops, and how cancer develop, and how it remain in the body, and how your immune system is not able to get rid of that cancer or tumor cell, because cancer has that, or cancerous cell has that property. They are able to hide themselves. They just hide from your immune cells, so that they don't get killed. That's their strategies. And how they hide themselves, they don't they have antigen on their surface, but they don't give the MHC. So MHC looks like this, if you can see my hand. So this is MHC. Basically, it has antigen, which is here, and this portion, my hand, is MHC. So they don't extend the MHC. They just show the antigen on their surface, and that's how they hide it, because T cell is not gonna kill the tumor cell if there is no MHC. So they don't display MHC, and that's how they they hide from your cells. So what we do in CAR therapy is basically we introduce another receptor which works even if there is no MHC. So it's just a modified type of receptor. That's what we introduce, and that's what we make in lab. And basically we modify the T cell so that it has another type of receptor which works even if there is no MHC molecule on the target cell. For example, on the cancer cell, even if there is nothing, still it can bind and still it can kill it. Is it clear?
Yes,
hello, ma'am.
Okay.
Hello, here
Excuse me, ma'am.
Yes. Hi.
Ma'am, there is one doubt.
Yes.
Ma'am, how the modified receptors work on the other body cells that it causes various side effects?
Sorry, say it again.
Ma'am, how the modified receptors work on other body cells that it causes various side effects?
Yeah, okay, so like one example I'm telling you. So how it attached is, there are antigens, right? on the target cells, there are antigen. And if I talk about antigen, I'm giving one example. It's very, you know, simple example, like CD19, you might have heard it. CD28, you might have heard it. Or if even if you haven't heard it, that's okay. You're gonna read about it in your you know, future studies, whatever you are going to do, you are going to study them. So like CD19, CD28, these all are antigens, and they are present on the surface of many cells. For example, even on B cells, they are present. So what does that mean? If an antigen is, so for example, the CAR T cell is going to attach on a cell which has CD19 or CD28 on its surface, and then it's gonna attach. And that's how the signaling starts, and you know, your T cell is going to kill it. Now that same you know, CD19 is present even on your B cells, you know. So B cells basically secretes antibodies. And what happens, so imagine a situation like your T cells, like your drug, you know, your modified T cell is killing your own cell. That's how it happens, because it has the same antigen, but still we use it. And why we use it? Because we have even if your B cell gonna die, that's fine, because B cell is important, but not more important than your life. Why you are going for CAR T therapy? Because you are dying, right? You have cancer and no other therapy is working on you. So that's when you choose CAR T therapy. And even though you know that CAR T cells are going to kill your B cells, still we choose it. Why? Because you know, there will be less number of antibodies in your body, but still it's okay, because we have other cells, like we have plasma cells and other cells which can give you antibodies that you may need. And T cell are there, and which are stronger, so they're gonna protect you much larger extent, in a much larger extent. So I would call the side effect, the cytokine release syndrome, is because there is a flood, we call it cytokine storm is there, you know, body. So if I go back here, I will show you something. Yeah, here. So okay, so whenever there is antigen which is attached to this this CAR, it's gonna give you signaling, and then it call all of its friends. Sorry, go back. Yes. So it call all of its friends. For example, you know, mast cell, and then dendrite cell, B cells, and cytokines, all, and then so many enzymes. They call everyone to come and collect on this site to kill it. You know, so, and that's why there is storm of cytokines. Sometimes it's too much. Like, it's not less, it's like too much. So, and that's how there is side effects, because there is so much fight going on, you know. So there is a lot of, you know, fever, high temperature, and then low blood pressure, hypertension, and you know, dizziness, and because a lot of thing is going on, like so many cells get activated. And here, you know, that if something is there, then your cells are, so basically your cells are dividing, and then each cell is calling its friend, you know, to fight with the cancer. So much things is going on, and that's why there is that side effect, like cytokine syndrome is there, storm is there, like too much thing is going on. So that's basically the side effect, which is kind of good thing, because it means that your CAR T is working. And let me tell you one more thing. If there is no side effects, I'm very sorry, but I have seen patients which don't show any any stronger symptoms like these type of CRS, like cytokine release syndrome. If you don't have it, doctors are very sorry, but they have to inform that CAR T therapy is not working on you. And I mean, it's very sad, but it happens that sometimes it don't work on you. And why it don't work, no one knows. Like few school of thoughts are saying that probably your CAR T is not able to, you know, combine with the antigen, and reason could be anything. Reason can be, for example, there is no CD19 on the surface, because the CAR T is made, for example, here this. Are you able to see my cursor?
Yes.
Okay,
Nice.
so here, this, for example, this thing is CD19, and this CAR is made specifically to attach on the CD19. But if there is no CD19 here, then this CAR won't be able to attach it, and then it won't be able to give the signaling, and then this T cell won't be able to call its friend and, you know, enzymes and other cytokines, and so it just fails. So that's how it happens. So sometimes it happens that even after giving this the T cell, the CAR T cells, there is no side effect. It means that nothing is going on. You know, it means that your cancer is still moving free, and it's not working, which is sad, very sad, but it happens. Okay, is it clear, your question? I'm not able to hear you.
Whoever had asked the question, you can just raise your hand. I'll just unmute you. I guess you can continue because I have. Okay.
I can see like 3 raised hands.
Okay. I'll just unmute. Alok, I'm just unmuting you if you want to have a question.
Hello, ma'am. This is Alok. I am a DST sir, project student at the Central University, Punjab. I have a question related to this. Actually, what you have done, you have just developed this technique specifically for leukemia only. For the time being.
Leukemia and lymph basically for
Means
lymphocytes, like for
only blood related cells cancers, right? No.
Yes, currently.
So,
But
as you are saying,
yes, continue.
Okay, as you are saying that some patients shows don't, like some patients after these getting them this card cell, they are not showing any kind of immune response, which basically they should show.
Yeah.
Okay, so you try to develop gene specific or this gene specific card cell.
We make antigen specific CARs, antigen specific CAR cells. And why antigen specific? Because like most of the leukemia cells have CD19. So basically, I know in my office we make based on CD19. But if there is some other type of, you know, antigen which is not included in your CAR T therapy, you know, cell production, then that won't work on this thing. And this is the future work that need to be done. Like if you see, I'll show you like few things later, which shows that in future, what all we can do. Like for example, currently, at least I'm not aware of that CAR T cell is working on the tumor cells. Few companies are working on that, and that companies are really hot in the market. Like for example, if I talk about Kite company, there is one more big company that's name is Guild, and Guild just acquired that small company, you know, because that company was going to get FDA approval, you know, within like I guess 2 or 3 days. After 2 or 3 days, it was going to get the FDA approval, and before like 2-3 days, the bigger company acquired it because it's very hot in the market, and we need that sort of treatment and that sort of I'm sorry to say, but that sort of money, you know. Because CAR T therapies are pretty costly if you know the rates. Like it's done, it's very costly. It's like $350,000 to $500,000. It's pretty expensive, but it is personalized medicine if you can understand that. So And why it's
Okay.
personalized? Because it's not like over the counter. It's not that you're gonna get on some what do you call it, like a store, like a medical store. You are not going to get it because it's personalized. You are it's your T cells that are getting modified. So it's, you know, each and every cost is yours. You have to pay for that thing. Even
Okay.
here it's very, very costly. So like our company here, if you talk about my, I was working with Juno Therapeutics, which was taken over by Celgene, and then now Celgene is taken over by Bristol Myers Squibb. I mean, like 74 billion US dollar, something like that, 78, I don't remember, but like it's, and Juno Therapeutics is going to get its FDA approval for CAR therapy pretty soon. So it's, so we all are like same now. Juno Therapeutics is Bristol Myers because it's taken over.
Okay. What type of T cell are you using it with, whether it is helper or cytotoxic?
So we basically use both type of TC as well as TH cells. Yes, we use
Okay.
both cells. Yeah.
Okay, okay, ma'am. Thank you very much.
Yeah, thank you. Okay, so I have a video of my my workplace, which shows what we do and how we work. So I would just like to open it.
How do you not get up in the morning?
Okay, so here, one more thing, sorry. Jump is, it says jump, right? So it's Juno Therapeutics manufacturing plant. So that's why it's called jump.
How do you not get up in the morning? I tell myself that every day. There's someone who can't get up. There's someone who is relying on me to go in and help them. There's someone who's wondering where their cells are today. Where are they in processing? Are they close to getting them back? They're looking at their watches. So how do I not go?
I have a personal vendetta against cancer. My mother had triple negative breast cancer, and she passed away when she was 64. It really focused my efforts and made me double down even harder to deliver these therapies to patients that need them.
I like being able to make a difference, especially with people who are suffering. Because my son had cancer when he was 9. We just rolled the dice, and we just happened to be the lucky ones. And I knew I wanted to join the battle against cancer.
Here at Jump is where we make the CAR T cells. We take the patient's blood, we receive it, we isolate the cells that we specifically are looking for, and then we re-engineer them so that they recognize cancer within the body. We grow them until it's reached a certain point, and then we harvest them and we package them out and send them to the patient.
I believe in what we're doing. I want the staff to be completely committed to bring the highest quality that we can bring to this production staff and get this product back to the patients.
I love the people I work with. I think we're all have the same mission. We're all invested and we all want to have the same goal. I just feel like we're really united and we're really focused on the goal.
Starting with my dad having cancer and then able to, study science and then eventually get a job at Juno where I'm treating the disease that he has. It's just, I'm just getting emotional just talking about it. It's really cool.
In middle school, my dad was diagnosed with cancer. He had a tumor on his kidney. Knowing that the work you're doing, that someone needs you to complete your work and that someone is putting their life in your hands, that's the thing that keeps you motivated through my childhood and to now. We can change the world here. That's what's so exciting about working here too. My name's Nick Nestingen.
I'm James Roy.
Judy Lau, Jackie Sweeney,
Dan Slater,
Yvonne Stevens,
Zachary Tremblay,
Wesley Starr,
Stephanie, Tana Shogi,
Bernaldi,
Alex Hannah Schultz, Sydney,
Terrence, Jeff Maston,
Adam Hughes,
Brittany, I'm Amanda Lee.
We make our T cells.
Okay, I hope you like it. These all were my colleagues, so I love it. Okay, so next. So this is the hospital which I was talking about, Fred Hutch, where actually our patient goes and where the drug is, administered to them. And this is the building, Juno Therapeutics, my office. This is the manufacturing plant. This is Celgene that I was talking about. We are under Celgene, and Celgene is under Bristol Myers Squibb. And these were the side effects that we were talking about. So let me minimize this thing, sorry. Yeah, okay, so these were the side effects, which are CRS. That's what I was talking about. CRS is cytokine release syndrome. So there will be fever, hypertension or hypotension, respiratory, and just fogging of brain and dizziness, seizures. Anything can happen, but it depends on person to person. And doctors are now very capable. There are medications, though. It's a good news that you can certainly handle these symptoms, and there are medication to subside or curb that side effects. And side effects is good. I mean, doctor get happy when this is probably the only time doctor get happy, when you get sick. So okay, so these are the bottleneck that I was talking about earlier. In one of you guys asked the question. So what are the bottleneck? What are the failure? Why it fails if they fail? So sometimes the cells are unable to harvest. What does that mean? That means, for example, we are introducing the CAR gene in the cell, and then we grow the number of cells because we need minimum quantity, like how much you are going to administer in a patient. So sometimes it happens that cells are not growing. So that's one type of failure. Sometimes it happens. And another thing is what exactly antigen you are using. For example, majority of time, like only 3 drugs are currently in market. Like 3 I'm talking about, including Juno Therapeutics, which is like going to approve very soon, like within a month or 2 maybe. So only 3 drugs are currently in market. So most of them are using CD19 and CD28 as their antigen, but there can be so many other type of antigen, and CAR T can be made using that type of antigen. So what antigen you are using? So better, research is needed. I mean, research is going on very well, but you guys are future, so you may end up finding another antigen which is present in different kind of cancer, maybe on tumor cancer or something. So what antigen you are using, that's what make CAR T successful. And then another bottleneck, I would not call it a bottleneck, but the problem, the issue is like toxicities, like neurotoxicity did happen like earlier, and there are just severe case of CRS. And another thing is B cell aplasia, which means because CD19 is also present on your B cells, so your CAR cells are also killing your B cells, which is not good, but you don't have any other option. And B cells are required for antibody production, but antibody can be produced by plasma cells as well. So you have like backup, but still this is an issue. And another bottleneck is like a lot of different type of cancer is not like currently at least not treated by CAR T, because it's the research is needed, like more research is needed. So it's like solid tumors, like central nervous system disease or lymphoma of other types are not. So what is the future of CAR T? So I already talked about it, like research is needed, like different research is needed, and different checkpoints are needed. What else we can use apart from CD19 and CD28, to block a tumor, or can we like have multiple, artificially synthesized CAR in the same cell? Anything can happen. There is, so many different possibilities. So let's see how it goes. That's all I have for today. So if you have any questions, you are welcome to ask.
Okay. You can just raise your hand here. Rashmi is having any question?
Double click.
Hello, ma'am.
Yes, hi.
You just raise your hand. I'll unmute you. There is Rashmi. Rashmi is having a question. Rashmi, you want to ask?
Yes, ma'am. I would like to know what is the success rate of CAR T-cell therapy in clinical development, because it is disease specific. I would like to know its success rate in clinical development.
Yes, okay, so I'm unmuted now. Okay, so success rate is pretty good. So currently CAR T, which is in market, is only for leukemia and lymphoma. These both have a pretty good, I would call, success rate, like 95% and 80% respectively. And a lot of other research is going on as well. So hopefully very soon we will come up with a number, but currently just for leukemia and lymphoma, since it's in market, so we know the correct number, which is like 80%, 95%.
All right, ma'am. Yeah, thank you.
Thank you.
Okay, Arvinder, ask your question.
Yes, ma'am. Thank you so much, ma'am, for this wonderful presentation. I'm Arvinder Singh. So my question is, you are using T cells for CAR technology, but T cell cannot generate memory. As you just said, the cancer just came back. Okay.
Yeah.
So instead of using T cell, why you guys don't use B cell? Because B cell can produce plasma cell and it can produce memory cells. So in future, if cancer just came to the cancer and just end up, you getting my point, ma'am?
Yeah. Okay. So why we are using T cell instead of B cell is it's basically we also use B cells. B cells and T cells both are used. For example, but B cells are difficult to handle.
Okay. Sorry, ma'am, I can't hear you.
Just wait. I think there is some net connection at her.
Okay, I'll wait. Okay.
She's not showing on the screen.
She is there, but I guess so. There is some internet issue. Yes. We can hear you, ma'am.
Okay. So yes. So I was talking about B cells and T cells. So T cells are easier to handle. It's easier to transfect a T cell rather than B cell, and also since our antigen that we are making does have a, CD19 is already present on the B cells. It's difficult because it's same thing both side, because your for example, I have B cell and then I have tumor, and both have same antigen. So you get it? It's difficult because
yes, ma'am. Yes, ma'am.
Yeah, because it's going to attach on its own B cells, because it also have the same antigen. B cells. I told you when we administered T cells, CAR T cells, it's gonna kill your B cells as well. And what you are asking me to do is like make my car on B cells. But B cells, don't forget that B cells also have CD19 on their surface. So who is gonna kill it? Like your car is going to kill your B cells. So it's gonna be a huge failure.
Yes, ma'am.
But
I got
question, I like it.
it. Okay, thank you. Thank you so much.
Mhm.
Any other question?
There's Sarthak. I'll just unmute Sarthak.
Hello, ma'am.
Hi.
Ma'am, my question is, ma'am, cannot we do some modifications in B cell to protect their CD19?
The one, and I would say that, another approach can be, instead of using CD19 as our antigen, we can use some other antigen. There are so many type of, CDs in the cells. So we may need, other kind of research which identify other target antigen. And if we can find it, then of course, yeah, we can use B cell maybe in future.
I can see a lot of hands. Jutika, Rashmi.
Yeah, go ahead. I love it.
So, Roma, unmute them one by one.
Okay, Harsh, you can just start your question. Harsh Sahu. Yes, Harsh. Okay.
Hello.
Hi. Yes.
Hello, ma'am. I'm Juthika. Ma'am, I was asking how are CAR T cells made, by recombination or by using those plasmids or any other type of process?
Sorry, your voice is breaking. Sorry, I'm not able to hear you. Can you repeat?
Jutika, you need to unmute yourself. I've unmuted from my side. Any other question? Harsh, you want to ask question? You can unmute yourself.
Hello, ma'am.
Hi. Yes.
Ma'am, ma'am, currently, how many countries are using this CAR T cell?
So CAR T is, okay, so India is certainly not using it. It's very, very costly, but I have a good news for you guys. Biocon, right? So Kiran Majumdar, and then there is a doctor here in USA who is collaborating with Kiran Majumdar and bringing this and trying to basically bring this CAR T therapy in India, not very soon, but hopefully very soon. So currently Europe has, I know about my company, we are in process of, doing this thing. So there are a few countries in Europe, and then US have it. Certainly these two have CAR T therapies. Mm-hmm.
So you want to take more questions?
Yeah, I have all night.
Okay, Shivani.
I mean, I don't know how much time you guys have, so it depends on your, I'm good.
Hello.
Yes, hi Shivani.
Hello, ma'am. Ma'am, my question is that what is the difference between targeted chemotherapy and CAR T cell therapy? Because in both the in processes we are actually focusing on the causing a cancer cell.
Yeah, so CAR T, so targeted is not anything about chimeric antigen receptor. It's the targeted is more about, you know, giving medication to that particular spot and trying to dissolve tumor or whatnot, but not with the help of chimeric, anything chimeric. So, so targeted therapy is nothing to do with what you call it, chimeric antigen receptor. So this genetic engineering thing is only with the CAR and not with the target.
Okay. Okay. Thank you, ma'am.
Thank you.
Okay, one more question I'll take from Ashwarya.
Hi, yes, Aishwarya.
Do you utilize synthetic biological approach for CAR gene sequencing?
Sorry, your voice is breaking. Sorry, can you repeat?
Hello, ma'am.
Yes, hi.
Are you using synthetic biology approach to generate the CAR gene sequence?
Synthetic what? Sorry?
Synthetic biology.
Synthetic, sorry?
Biology. Approach.
Can anyone repeat what? Roma, can you repeat the question?
Actually, her voice is not clear. Can you just repeat,
you?
Ashwarya, a bit loud?
She was actually asking about synthetic biology.
Synthetic, if you call it synthetic biology, I won't mind calling it synthetic biology, but it is definitely not natural, like we synthesize in lab. CARs are synthesized in lab. It is artificial. So if you like to call it synthetic biology, it is synthetic biology. But we, with the help of gene therapy, immunotherapy, we make CAR cells, and yes, it is synthetic. So yes.
Okay, ma'am.
Okay, I think Roma, we don't have much time to take the questions now.
Okay.
Thank you, Pooja. Thank you so much.
Thank you so much, ma'am.
It was new for me, you know. I learned from you today that yes, we can use chimeric antigen receptors on T cell and how we can utilize it for immunotherapy and what is the success rate.
Yeah.
And also you shared drawbacks. That was very interesting. It enhanced my own knowledge. I came to know about a new technology. And,
thank you so much.
And we are very grateful to you. How is it going? Because amidst the COVID infection and that to its highest in USA, we are so much grateful to you for taking out the time. How is the situation?
Yeah. You know, so the first infection was here in US. I'm talking about the first infection was in my city, like here in Seattle. So we all are working from home. Me and my husband. I have a son of 1 year old. So we all are, you know, at home. Like it's not completely like lockdown like back in India. It's completely lockdown, I heard, but it's like more stay at home order kind of thing. So we are just staying at home and you know, just enjoying my time with my son because otherwise I just go to office and he goes to, you know.
I know. So take care, stay safe, and we wish everything becomes fine. Thank you.
Yeah, hopefully soon. Yeah.
Thank you
so
for
much.
taking your time.
Yeah,
Thank you.
so much. Yeah, thank you. Bye, everyone. Thanks, Roma.
Thank you so much, ma'am.
Thank you. Bye.
Bye.
I'll talk to you offline.
Sure.
Okay. Thank you.
So now we have with us our next speaker, Professor M.V. Rajim. Good afternoon, sir. Unmute kar do, Roman. Sir is not.
Good afternoon. Good afternoon, everyone.
Good afternoon, sir. So
I'm
we welcome
audible?
you on the forum of SAP Bioescalator.
Thank
Thank you for accepting our invitation, sir. Thank you. I request Dr. Abhinav to please introduce, sir.
It's my great privilege and honor to welcome Professor M.V. Rajam in the platform of SAIP, Bioescalator Insight into Biotechnology, Plant Sciences and Bioinformatics. This is a 4-week program, sir, and Professor Rajam has earned his master's and PhD in botany from Kakatiya University. Professor Rajam has a rich experience of working in different domains of plant biotechnology and RNAi. and development of crop plants for disease and pest resistance, as well as important improvement of other agronomic traits using RNA and microRNA interface strategies. Professor Azam has more than 30 years of teaching experience, and he's a fellow of all the 4 academies in our country. Professor Rajim has completed more than 30 research projects, funded research projects from different agencies of our country, as well as from several companies and collaborative projects as well. Professor Rajim has 3 patents, and he guided more than 30 students, PhD students, and several MPhil students as well. More than 25 postdoctoral fellows is being trained in his lab, and he trained more than 150 faculties in his lab. Professor Azam is one of the pioneer, is one of the biggest name of plant biotechnology in our country. I welcome Professor Azam to express his pearl of wisdom, to express his lecture as a debatable lecture, I must say, and it's a privilege and an honor to welcome Professor Rajim again. Thank you, sir, being a part of SAIP.
Thank you, Abhinav. Yeah, just a second. Yeah.
Yes,
Full
sir, we
screen
are able
able?
to see.
Okay. Yeah. Am I audible?
Yes, sir.
Yeah, okay.
Yes, sir, you are audible.
Yeah. Yeah. Again, thank you, Abhinav, and your colleagues for giving me this opportunity to talk about the applications of plant biotechnology in agriculture. So I will be covering basically the concepts of plant tissue culture and applications, and as well as plant genetic engineering and potential applications, including RNA interference. As you all know, the food problems have haunted mankind since time immemorial. And in 1960s, the hunger, malnutrition were widespread, especially in Asia, including India. And more than 800 million people cannot afford 2 courses of meals, and about 30,000 people, half of them are children, they suffer due to hunger and malnutrition. More than 1 million people live on less than $1 a day, which is really amazing. And as you know, the today's population size is about 7 million, and it's going to be more than 9.7 billion by 2050. So therefore, there's a great demand for food production to feed the growing mass. In the next 50 years, mankind will consume as much as food as we have consumed since the beginning of agriculture, that is about 10,000 years. This was said by Dr. Clee James, who has been updating about the cultivation of genetically modified plants globally. The agriculture, not only in India, globally, it is suffered by many issues or problems, particularly the declining of crop productivity, diminishing of grain production, the sinking water resources, and also growing population. These are some of the, and also the croplands are quite vulnerable for abiotic and biotic stresses, which significantly affect yield. So therefore, we have to crop improvement for yield improvement is absolutely important. And looking at the population size and food production, we have to improve our croplands by more than double by next 40 or 50 years by using conventional plant breeding as well as the biotechnological approaches. In 1970s, Dr. Norman Borlaug, he has developed the improved varieties of wheat, particularly semi-dwarf and dwarf varieties by deploying dwarfing genes from Japan. And this was responsible for first green revolution in 1970s, which really greatly improved wheat yields, not only in India, in the Pakistan and elsewhere. Then in India, Dr. Swaminathan is regarded as architect of green revolution in India. He is also responsible for the improvement of not only the wheat and also rice. And Dr. Gurgovind Kush from International Rice Research Institute, he has developed quite a few rice improved varieties, hybrids. And in fact, more than 60% of the improved rice hybrids were virtually developed by Dr. Kush. Uh, and more recently, he has also come up with the super rice, which is resistant to biotic and abiotic and also adapted to a wide variety of environment. So what I'm trying to say is plant breeding has contributed very significantly for crop improvement, and more than 90%, even 95% of the improved varieties were virtually developed by plant breeding methods. But the plant breeding, it has its own limitation. Usually, it takes about 12 to 15 years to develop a new variety, and we can only mobilize the genes between closely related plants. We have no way to transfer a gene between distantly related plants because of sexual incompatibility. So that means there is a limited gene pool in case of plant breeding, and the gene transfer is also not precise. Along with the useful transfer of useful gene, we will also be transferring many undesirable genes through crossbreeding. So in order to get rid of those undesirable genes, we have to subject the F1 hybrid for back crosses, 4 to 5 back crosses. So this is time consuming. And all these limitations can be overcome by genetic engineering methods, right? So you may ask, why we should go for plant biotechnology, in particular plant genetic engineering or transgenics. See, it has been projected that there's an increase in global demand, between 1993 and 2020, about 40%. We need to increase about 41% in case of cereals, as well as roots and tubers. And more recently, I've read an article where it was said that 70% increase in food production is required by 2050 to feed 9.1, actually 9.7 billion world population, uh, according to FAO 2009. And some countries, particularly the South Asian or Asian countries, including India, we are at a high risk for food demand. If you look at the food production versus population, there's a huge gap between these. And unless we bridge the gap between food production and population size, we are going to face great famines in the coming years. So therefore, the doubling of a food production, that too sustainably on the available arable land, is absolutely important for increase of the food production. So if you look at the technology options, we have 5 different options, but more importantly, plant breeding and genetic engineering is absolutely important. So we have to couple plant breeding methods with the genetic engineering or transgenic technology to meet such kind of high demand, about 70% increase in food production. So looking at these figures, definitely we need a second green revolution to happen. In fact, in the last Indian Science Congress meeting, our Honorable Prime Minister Modi ji had also emphasized about need of a second green revolution. I'll start with the role of plant cell and tissue culture for crop improvement. The plant tissue culture, plant regeneration, that is regeneration of whole plants, cultured parts or explants like stem piece or leaf piece, is absolutely required for development of genetically modified or transplants. The plants have a very unique property, that is totipotency. Unlike animals and humans, where only the fertilized zygote is cellular totipotent. We have no way to produce the whole organism in case of animals and humans other than fertilized zygote. But in case of plants, virtually you can regenerate plants from any plant part. Basically, the plant tissue culture regeneration involves the collection of an explant. It can be a piece of leaf or stem or any plant pot, and then culture the plant pot explant on a suitable nutrient medium in tissue culture vessels like test tubes or germ bottles. So after inoculation of plant pot on a suitable nutrient medium having various nutrients, macro, micronutrients, as well as phytohormones like auxin, cytokinin, we, initially we may get callus, and this callus can be utilized to regenerate shoots or somatic embryos by changing the nutrient medium in order to have complete plants. Uh, so this is, the basic steps involved in plant tissue culture. If you look at, or if you compare plants versus animals with regard to cellular pluripotency, I've already mentioned, like in case of only fertilized zygote is 30%, but the stem cells in case of animals and humans, they are pluripotent. They have, you can regenerate various organs or various kinds of cells, different lineages from the stem cells. But some optimistic scientists, particularly from US, some years back, they think that pluripotency may be possible even in case of animals and humans. So therefore, they started preserving human bodies for future life. If tomorrow somebody discovers the right factor or factors which can be involved in regeneration of whole organism from cultured somatic cells of animals and humans, so it would be possible to regenerate the whole animal or human from stored or preserved human body if this happens in the future. But you know uh, fortunately it has not happened because we already have a population problem, so this may not be ethical. Actually, there will be a lot of ethical problems with this. But on the other hand, the cloning of plants or regeneration of plants, it has many potential applications in agriculture. So we will discuss all those. uh, for the first time, Dr. Gottlieb Heberlein from Germany, he made the first attempt to mechanically isolate and culture single cells, but unfortunately he was unsuccessful. His cells could only increase in size, but his cells were never divided. The reason was very simple. So those days in 1902, the sterilization techniques were not available. The nutrient medium was very simple. The hormones were not discovered. So because of these reasons, he was very unsuccessful. But Dr. Heberlein, he had a great foresight. He could predict several things in plant tissue culture. Like he said, the formation of artificial embryos, uh, is possible from tissue cultures, and also use of coconut milk in plant tissue culture would be useful for cell division and growth of the cultured plant pots. And all these have been experimentally proved or demonstrated in subsequent years. The plant tissue culture media, there are more than a dozen different nutrient media, but the one nutrient medium which is called Murashigi and Skoog or MS medium, which is most commonly used for culturing plant tissues and cells. And the medium, it has several constraints like inorganic salt formulation, both macro and micronutrients. The source of carbohydrate, particularly we use sucrose. Rarely we also use maltose as a carbon source. Then vitamins, water, and also the more importantly plant hormones. There are 5 different phytohormones, but excuse me. Yeah, sorry. So particularly we use the auxins and cytokinins for the regeneration. And we also use solidified agents like agar for solidifying the medium. For cell cultures or protoplast culture, we use liquid medium without solidified agents. So these are the two guys, Professor Skoog and Dr. Murashigi, they are the one who has developed the most commonly used plant tissue culture medium, MS medium, popularly known as MS medium. And this medium is quite suitable for culturing, any plant pot, including anthers, protoplast cells, and this is suitable because it is highly rich in nitrogen source as compared to Yeah. Can you hear me now? Yeah. Okay. Oh. Yeah. So the tissue culture medium and the tissue culture vessels, they had to be
So the screen
sterilized before
sharing
they.
is not on.
Oh, okay. Prem, just a second.
Okay, sir.
Hello, Prem. Prem.
So there is this icon share screen. You can just click it
Pardon?
and there is this icon share screen. You can just
Yeah,
click
but
it.
I could not see the icons. I'm not quite familiar with this.
It's okay,
Just
sir.
a second. I'm calling my son-in-law.
Okay, sir.
Prem. Hello, Prem. No icon. I could no. I can't see icons.
The
I don't know
icons
what's
are
happening.
on the home page of Zoom, right below.
Yeah, the below I can't see those actually. Hmm. Mhm. Oh, okay. Yeah, now I can see. Yeah,
So you just need,
yeah.
okay, sir.
Okay.
Yes, sir, it's
Yeah.
visible.
Is it okay, or should I go for slideshow? The screen is okay now.
Yes, sir, that is visible.
Okay. Yeah. Yeah. So the tissue cultures they had to be maintained in a growth chamber or walk-in environment chamber, or we call it as a plant tissue culture room, where we maintain more or less constant temperature, about 26 degrees Celsius, and light intensity as well as dark and light cycles and relative humidity. So these are absolutely required, optimum conditions of these required for culture of plant cells for regeneration. And as I said, plant hormones, particularly auxin cytokinin, they play a pivotal role for differentiation of plant cells into either adventitious shoots or somatic embryos. See, basically, the ratio between auxin and cytokinin is most important. If your nutrient medium, if it has high ratio of auxin to cytokinin, mostly we end up getting root development. And if medium has high concentration of cytokinin to auxin, mostly we get shoot regeneration. If they are present in moderate concentration, we get callus formation, that is undifferentiated cells. So for plant regeneration, the manipulation of phytohormones is absolutely important. The other ingredients of nutrient medium is more or less same in MS medium. So, so these are the different steps involved in direct organogenesis, which is mostly used for clonal micropropagation or propagation of elite plants through plant tissue cultures. Like this, this is from our own lab work. In case of citrus, sweet orange. So we germinate the seeds on a nutrient medium in test tube, and then we collect epicotyl segments as explants, culture them on shoot regeneration medium. After about 3 weeks or so, you can start seeing the induction of shoots, either from one end or from both ends. And then the developed shoots, they can be individual shoots can be excised out and transferred onto a rooting medium for regeneration of complete plants, which can be later established in pot condition and then later in greenhouse or net house. So that was direct shoot regeneration or organogenesis. And depending on the phytohormones or concentration of phytohormones, sometimes initially we get a callus, and from the callus can be manipulated to get advantaged shoots. So this pathway is called indirect organogenesis or indirect shoot regeneration. And then the developed shoots can be rooted in vitro, and then regenerated plants can be established in pot condition. And again, depending on the phytohormones, we can also induce the somatic embryos, the embryos from somatic cells, cultured somatic cells. And again, this has two pathways, direct or indirect somatic embryogenesis. If you get somatic embryos directly from cultured explant, so it is direct somatic embryogenesis. And if you induce somatic embryos from the intermediate callus, so it is called indirect somatic embryogenesis. So the somatic embryos, they are similar to seed embryo or zygotic embryos, which has three different stages: the globular and globular embryos, heart shape, and torpedo or coronary embryos. So these are the two different pathways of plant regeneration. Both can be utilized not only for micropropagation or for development of genetically modified plants. The cell and tissue culture, they have potential applications in agriculture. For instance, we can propagate the elite plants, in huge number. Right? It is possible to produce millions of plants, in a year's time, even less than a year, through micropropagation or tissue culture techniques using the nodal segments or shoot tip or meristem as starting material. And it is also possible to develop the virus-free plants by culturing very tiny meristems. And as you know, many of the plants, they also produce the pharmaceutically important secondary metabolites. For example, the taxol, which is being extracted from taxus, this is a very precious compound because it has anti-costin property. And these days, the taxus and many other medicinally important secondary metabolites which are being produced from cell and tissue cultures. The plant tissue cultures, particularly the protoplast cultures, can be used to develop the somatic hybrids between sexually incompatible species. It is very difficult to produce hybrids between distantly related plants. For example, two different species of a genera or two different varieties of a species. So very difficult to cross because of sexual incompatibility. In those cases, it is possible to produce raised somatic hybrids through protoplast fusion technology. Here, we take protoplasts of two different plants and then fuse them using fusogenic agents or electrofusion. Then the protoplasts are fused. The fused product or the hybrid cells can be cultured on suitable nutrient medium in order to produce the somatic hybrids. This is one of the most important applications of plant cell culture technology. Like Dr. Melchers from Germany is the first one to make an attempt to produce an intergeneric somatic hybrid between potato and tomato. As you know, it would be not possible to cross potato and tomato because they belong to different genera and because of sex incompatibility. But he made an attempt to fuse the protoplast of tomato and potato in a view to produce an intergeneric somatic hybrid. He could get a hybrid which he named as a tomato or tomato, depending on the characteristics. But unfortunately, the intergeneric somatic hybrid between these two plants was not normal because of selective elimination of chromosomes from hybrid cell. But this has remained as a future dream. It may be possible to produce the viable somatic hybrids between two different genera like tomato and potato by a different technique called asymmetric somatic hybridization. And if that happens, it would be boon in agriculture. You can have two different kinds of fruits or even three different kinds of fruits through asymmetric protoplast fusion technology. Again, as I told you, the distant crosses, interspecific and intergeneric crosses are not possible because of sexual incompatibility. But here is very very simple technology called embryo rescue. In general, in case of distant crosses, we do get seed, but seed is usually sterile because of aborted endosperm. And in such cases, you can excise the embryo, and the excised embryo can be cultured on suitable medium to regenerate plants. And these plants are nothing but hybrids, very novel hybrids, which are not possible, otherwise not possible through plant breeding methods. So the embryo rescue has been successfully employed or used in case of mustard and few other crops for development of novel hybrids. Similarly, there's another technique called in vitro pollination and fertilization. This technology was developed by Professor Panchan Maheshwari from Delhi State Botany Department. And this technology can also be used to develop novel hybrids between distantly related plants. And he could develop the rare and novel hybrids in few plants like petunia different species, and you could even get seeds in test tube through in vitro pollination and fertilization technology. Then again, the Satish Chandra Maheshwari, the son of Panchan Maheshwari, along with his colleague, Professor Shipra Guha Mukherjee from JNU. So they were the first to discover androgenesis, that is development of hoplite plants from cultured anthers. And this has really revolutionized the whole agriculture, because hoplites can be used as for doubling of through doubling of chromosomes. You can develop genetically homozygous plants which can be used as the starting parents in crossbreeding. And also there are a couple of other important applications. For mutations can be easily expressed in hoplite cells, and one can also produce double hoplites through anther culture of F1 hybrids, and these have very, very important to develop recombinant inbred lines. And tissue culture technology can also be used for many other applications, like seedless plants through endosperm culture and many other applications. Then now we will come to the role of plant genetic engineering crop improvement program. And as I said, plant breeding basically it relies on genetic variation, which is already present in the nature. Look at here. In case of beans, you have all kind of variation in seeds. You have different sizes, different colors, and different shapes. And you may ask, when we have such great genetic variation in nature, why we really need plant biotechnology or transgenic technology? So, that's because the nature does not contain all the genetic variation that we require. For instance, a banana, genetically modified banana, which can produce an antigen against a dreadful human diseases like cholera. So this is not possible by nature, but it is possible through genetic engineering. So similarly, the golden rice, golden rice was created to have rich the beta carotene of provitamin A, and later, of course, they have also modified it to have uh, iron rich and iron absorption. So this is not possible by nature, but we can create these kind of plants through genetic engineering. If you ask my choice, I would love to have genetically modified strawberry with blue color, right? Which is absolutely possible by genetic engineering. So therefore, all these in points which imply the need of the biotechnology for crop improvement program. The concept of genetic engineering is not new. Look at the earlier perceptions of engineered plants. On your left panel, there's a flower looking like lion, and the middle and right panel, you have men and women having a twig on their heads. So these actually, the left one, this is a plant-tiger combination, which was published in a book called A Book of Nonsense, published in 1862. The middle and the last one, a plant-human combination, male and female mandrake. So this was also published in a book about 1498. So you can imagine if this, of course, this may not be possible, but somebody has tried to fuse an animal cell, that is erythrocyte cell of a frog with the tobacco cell. He could get hybrid cell, of course, for obvious reasons. He could not regenerate whole organism. But for a moment, just think if it is possible in the future, it would be possible to produce these kind of humans, a human having a plant twig on their head. If that happens, the humans will become autotrophic. We can synthesize our own food. You know, this is only future possibility. Yeah. So as I said, plant genetic engineering has many potential applications in agriculture, for crop improvement. Then genetic engineering basically involves an insertion of a useful gene, one gene, even more than one gene, into crop plants to have a desirable trait. It can be a disease resistance or insect resistance or yield improvement, etc. And I've already mentioned the genetic engineering has quite a few advantages over plant breeding. So, and virtually we can improve any agronomical important character through genetic engineering. So the biotechnology, basically we have three different kind of biotechnology. Agri biotechnology or plant biotechnology basically deals with plant cell and culture and genetically modified plants. Then medical biotechnology, industrial biotechnology, and here of course I'm dealing with agri biotechnology or plant genetic engineering. Initially, I'm just showing some of the products synthesized by bacteria, genetically modified bacteria. In fact, the human insulin for diabetics was the first biotech product which is being produced in genetically modified E. coli or bacterium. And of course, it is being practiced to develop chemicals which can block HIV infection, or through bacteria, it is also possible to produce recombinant proteins or biodiesel fuel or bioplastics, that is production of polyhydroxybutyrate, which is commonly used for the preparation of bioplastics. But these, the production of these compounds, there's a limitation. There's one great limitation. It is the use of bacteria is not cost effective for the production of these compounds. So therefore, the genetically modified plants, which can serve as bioreactors or factories for the production of a variety of useful products. So I will show you how the genetically modified plants can be produced and their applications in agriculture. How we make a genetically modified plant. Initially, first and foremost important is you have to have a beneficial or useful gene in hand, and this gene can be, the coding sequence of the gene can be cloned in a plant transmission vector. Most commonly, these days, most commonly we use binary plant transmission vectors based on the TA plasmid present in a bacterium called Agrobacterium tumefaciens, and the gene coding sequence of useful gene, it can be cloned in this vector, plasmid vector, under the control of an appropriate promoter and a terminator signal. And then along with the useful gene, we also clone a marker gene, usually an antibiotic marker gene, for the selection of transformed cells and to eliminate many thousands of untransformed cells. Then after introduction of or transfer of the gene into the plant cells are selected on selection medium, having lethal concentration of the antibiotic, and then the transformed plants, they can be established in pot condition. Then later on, we can verify the transgene integration, the integration of the foreign gene in the transformed plant by certain molecular biology techniques, particularly PCR, or southern blot hybridization can also be used for demonstrating transgene integration. And transgene expression can be checked by northern hybridization or quantitative PCR technique, right? And transgene protein can be checked by Western hybridization or the ELISA technique. So for gene transfer, there are many gene delivery systems in plant, but these days we use only two different gene transfer methods. One is based on the Agrobacterium tumefaciens or Agrobacterium rhizogenes. This is indirect or vector-mediated gene transfer. Or one can also go for gene gun or particle gun, which is a direct gene transfer. So in the plant transmission through agrobacterium, what we do, initially we collect a leaf disc from a leaf of about 1 month old. And then these leaf discs are infected with overnight grown agrobacterium cells having the gene of your interest. Then the infection is done for about 10 minutes. And then the infected plant tissue or cultured on shoot regeneration medium for a couple of days. And this step is called co-culture or co-cultivation. And during this time, the agrobacterium faithfully introduces the useful gene into the plant cells. I forgot to mention the agrobacterium tumefaciens is used for transmission because this bacterium is very unique. It has natural ability to transfer part of its plasmid, that is plasmid, the into plant cells at the infection site. So the agrobacterium is called natural genetic engineer of plants, and this organism is quite exploited for delivering genes into plant cells. So coming back to the second step, co-culture. After co-culture, the co-cultivated explants are now cultured on selection medium, having the lethal concentration of antibiotic, for instance, kanamycin resistant gene, that is neomycin phosphotransferase, which confers resistance to kanamycin and neomycin. So kanamycin can be used as a selection agent for selecting the transformed plant cells and eliminating many untransformed cells. So on selection medium, whatever the regenerants, that is shoots that you get, basically they are genetically transformed. And these transformed shoots can be rooted in vitro, and then the complete plantlets can be established pot condition and utilized for molecular analysis to show transient integration and expression. And then plants can be tested for the trait that we have introduced. For example, if you have introduced a gene for insect resistance, the developed transformants can be tested for insect resistance initially under laboratory conditions, and later on they can be tested under a field condition. And if they perform well, then they can be released as a new variety or transgenic variety. Alternatively, one can also use gene gun, which is also known as particle gun, microprojectile gun, or shotgun. I would prefer to call it as a gene gun because it exactly works like a usual gene gun. Usual gun, like here, you have a standard gene gun, right, which is helium driven. The DNA containing the gene of interest is basically precipitated on the surface of a very minute gold particles, and then these particles are called microprojectiles, and these microprojectiles can be shot or bombarded into the plant cells using the gene gun machine. More recently, one company, they have come up with a very small size gene gun, which really looks like a usual gun. Virtually, you can shoot genes into plants, even intact plants, by this method. So this is a case of direct gene transfer. So after shooting or bombardment of genes, you can select the transformed plant cells on selection medium, like I've discussed in case of agrobacterium, and that is how one can generate genetically modified plants, either through agrobacterium method or gene gun method. So the agrobacterium, most people prefer agrobacterium because it is very easy to perform, cost effective, right? As compared to particle gun, which is expensive because you may have to pay more than 1 lakh for 500 shots. But the particle gun is still used in those plants which are recalcitrant for regeneration. Let us discuss some of the potential applications of transgenic plants in agriculture. In essence, plant biotechnology, they provide us more food, better food in terms of nutrient quality, and better for the environment, because genetically modified plants can also be used for cleaning the bad environment, like a polluted soil with heavy metal, etc. And so far, till now, more than 50 important crop plants, genetically modified crop plants or biotech crops, that have been approved for commercialization in about 28 countries, including United States and India. Mostly, these crops include canola, corn, cotton, papaya, potato, etc. So There are a variety of benefits of genetically modified plants. For instance, we can increase crop yield. We can develop crop plants for resistance against pathogens and insect pests. We can increase nutrient quality or oil quality. Uh, you know, we can also produce edible vaccines and drugs in GM plants. Virtually, as I mentioned before, we can improve any agronomical important characters through genetic engineering technology. Uh, as you know, the BT cotton, which is resistant to a cotton bollworm or Helicobacter armigera, and this insect pest is quite notorious because the farmers, they use pesticides or insecticides very extensively to control this particular insect pest, very deadly insect pest. So for the first time, the Moon Center company from US, they have come up with BT cotton by where they have introduced a gene called BT gene or Cry gene in specific for engineering resistance against insect pests. Initially, they have come up with Volgard 1, where they could introduce only one gene, Cry1AC, for resistance against Helicobacter. But more recently, because of insect resistance problem against BT protein, they have introduced a second gene, and these cotton plants are called Volgard 2, where you have two genes to manage insect resistance problem. And the BT cotton, right, having cry genes, they are quite protected from insect pests. You can see yourself on your left, the non-transgenic cotton, which is badly affected by insect pests. On the right panel, you have genetically modified or BT cotton, where you have a lot of yield. Interestingly, like so about 15 years ago, we used to import cotton from neighboring countries, but now, because of increase in cotton yield by about 35 to 40% by cultivation of BT cotton hybrids, we now export cotton to other countries. So that is the potential of genetic engineering in agriculture. And more recently, the Myco Maharashtra Hybrids is company from Jalna, Maharashtra. They have come up with BT Brinjal hybrids, and also Indian Council of Agricultural Research, particularly Indian Agricultural Research Institute in Delhi. They have Dr. Anand Kumar and their colleagues. They have also developed BT Brinjal. So BT Brinjal is resistant to another most important insect pest, Lucinodus arbinola. It's called shoot and fruit borer in brinjal. In fact, the farmers, they extensively use insecticide for the control of this insect pest, and in fact, they give more than 40 times the spray, insecticide spray, for the control of this single insect pest. So BT Brinjal was approved by a highest apex body, that is GEAC, Genetic Engineering Appraisal Committee. But unfortunately, the BT Brinjal was not commercialized because the approval was not given by the government. And but recently, about 4 years back, the Myco company, they have transferred BT Brinjal to our neighbor country, Bangladesh. There, the BT Brinjal hybrids are performing so well, the farmers are quite benefited by cultivation of BT Brinjal. It's very unfortunate. BT Brinjal was developed in India, but it is not being commercialized in India till now. So in the future, in the near future, we may have BT Brinjal for commercialization. Likewise, we have many other potential applications of plant genetic engineering, like one can engineer virus resistance in some of the important crop plants like papaya, cassava, and other crops. I'll take a minute to explain why virus resistance is important in crop plants. As you know, every crop is badly infected by a variety of viruses. In case of papaya, papaya is very badly affected by a single virus called papaya ring spot virus. In fact, these papaya cultivars are extensively cultivated in Hawaii Island in US. About 15 years back, the farmers was losing most of the cultivated varieties because of the single viral infection. Most of the important varieties are already, extincted. At that time, the Dr. Roger Beachy from US, he has come up with a new idea. It's called cross protection. He could introduce the target same viral gene called coat protein into tobacco, and now the genetically modified tobacco expressing coat protein of tobacco mosaic virus is resistant to the same target virus. So this concept was used to develop the genetically modified papaya for resistance against papaya ring spot virus. So by deploying a single gene of the same target virus, now we could save the papaya from Hawaii and elsewhere. You can see yourself the right panel photograph where you have non-transgenic papaya. There's absolutely no fruits. On the right hand side, you have genetically modified papaya where you have a high yield, and there are no symptoms of virus in GM papaya. Similarly, the private companies like Monsanto, they have also come up with herbicide-tolerant canola, cotton, and few other soybean. And here they have introduced a gene called EPSP synthase from agrobacterium, and these plants are resistant to an herbicide, glyphosate. The glyphosate is very extensively used in developed countries like America for the control of unwanted weeds. And here, the canola, the plants, they are engineered to have resistance against the glyphosate herbicide. You can see on the left panel, the canola plants are badly affected by unwanted weeds in the field. On the right hand side, you have, after spray with the glyphosate herbicide, only the genetically modified canola is protected, and unwanted weeds were selectively killed. So I've given three important examples where by deploying a single gene, you can improve crop plants very significantly with these traits. And about 15 years back, golden rice was created by two scientists, Dr. Ingo Petrakis for Switzerland in collaboration with Dr. Bayer from Germany. And we scientists, we believe that golden rice is a pioneering example of 20th century because, and this is also a best example for multi-gene engineering because these scientists, they could introduce three genes involved in the whole biosynthetic pathway leading to the provitamin, that is beta carotene. So they could introduce three genes like phytone synthase, phytone desaturase, and lycopene beta cyclase. So introduction of the whole pathway, beta carotene pathway into rice. Why, you may ask, why they have created golden rice? Because the rice basically lacks beta carotene. So for obvious reason, the rice eaters, in fact, more than 50% of the world population, they rely on rice as a staple food. In other words, rice provides more than, calories for more than 50% of the population. So unfortunately, the rice eaters, they are deficient in vitamin A because rice lacks beta carotene. So because of this reason, they have engineered the whole pathway leading to provitamin A or beta carotene. So this is, it is named as a golden rice because golden rice grains, they look like golden color. Golden rice was developed about 15 years ago, and which was sponsored by, this project was sponsored by Syngenta, a multinational company from Europe. But unfortunately, golden rice has not yet been commercialized because of resistance from NGOs and other, some organizations. And but more recently, the golden rice seed was given to some of the developing countries, including India, to integrate the golden rice character into the Indian cultivars. In India, we have got golden rice seed through Department of Biotechnology, and this seed is given to three institutions in India: Indian Agricultural Research Institute, Coimbatore Agriculture University, and Indian Institute of Rice Research in Hyderabad. So they have already introduced golden rice character into the most popular cultivars of rice in India. So I'm quite hopeful that golden rice will be commercialized in India and other developing countries very soon. I've already mentioned about this. And Dr. A.K. Singh from IRI is the coordinator for this program. Yeah. So likewise, we can engineer many other important traits like, the delayed ripening in tomato was developed about 15 years back in US. As you know, the over ripening is a very important because farmers they lose tomato fruit because of over ripening. So therefore, the delayed ripening is important for longer shelf life of tomato. And here, you may be knowing that ethylene, the gas hormone, is involved in fruit ripening, and there are only two enzymes involved in ethyl formation: ACC synthase and ACC oxidase. So here, what they've done, they have used gene silencing technology called antisense RNA to knock down the expression of either ACC synthase or ACC oxidase to have a delayed ripening tomatoes. And these were named as flavor saver. In fact, flavor savored tomato, delayed ripening tomato, were first commercialized in US about 15 years back, right? And then later on, of course, now we have more than more than 20 important crop plants which have been commercialized globally, including BT cotton, etc. So the genetic engineering can also be used to improve oil quality, in terms of monosaturated fatty acids, and in fact, canola, soybean with improved oil quality have already been commercialized in many countries. Uh, People have come up with early flowering by introducing meristem identity gene. Like here, look at here, the citrus, in case of citrus species, usually takes about 5 years for flowering, but by introducing a single gene, meristem identity gene, they could induce flowering in just about 6 months. So this is very important because you can reduce the breeding cycle. Instead of waiting for 5 years to have flower and fruits, you can have fruits within 2 or 3 years. Similarly, people have developed seedless tomato in naturally colored cotton, like blue cotton, blue roses, abiotic stress tolerant transient plants, etc. There's another important application of genetic engineering. This field is called molecular farming or biofarming, where the genetically modified plants are being used as factories to produce a variety of useful compounds like anti-carcinogenic compounds or recombinant proteins, which are being used for many diseases, curing of many diseases. More recently, one US company called Genentech, they have developed a genetically modified corn, uh, which express a molecule. They did not reveal what kind of this molecule is, but this molecule can trigger the burning of stored fat. So obviously, the products like products that are made from this genetically modified corn, if you eat them, you can become physically or svelte physique or physically, you can burn your stored fat. And as you know, the obesity is a major problem in many countries. So these kind of genetically modified plants are very useful to take care of obesity problem in the future. As you know, the usual plastics are banned in all countries, including India. So therefore, these days, we get bioplastics, and the bioplastics are synthesized from a molecule called PHB, polyhydroxybutyrate. So, and this compound is being produced in genetically modified bacteria. But as you know, the using bacteria is not cost effective. So scientists, they're trying to produce polyhydroxybutyrate in genetically modified plant. Initially, they have engineered the whole pathway leading to polyhydroxybutyrate by introducing 3 different genes, and this was done in a model plant. You know, Model plant, but the PHB, the content was very low. Later on, they have expressed all these 3 genes chloroplast background, and they could improve 100-fold of this particular compound PHB. But what I would say, the introduction of genes for PHB synthesis is important in other plants like rapidly growing plants, tree plants, for example, so that you can extract this compound from leaves, without sacrificing the plants. There is another important application you may not be knowing, that a spider silk, this spider silk protein, which is called biosteel, and you won't believe if I say that spider silk protein has greatest mechanical strength among all natural and synthetic fibers. And this is a bioproduct and eco-friendly. It can be reusable. In fact, spider silk protein is being used for the preparation of bulletproofing, In fact, bulletproofs which are made from spider silk protein are much, much better in terms of quality as compared to the usual bulletproofs. And this protein is also used for the preparation of parachutes, ropes, optical fibers, etc. Another important application would be growing plants or bioluminescent plants. As you know, the insects, certain insects, insect firefly, they show bioluminescence or they glow during night. Similarly, there are some bacteria and some other organisms, they also show glow or bioluminescence. So for the first time, the Lux gene from insect firefly was introduced into tobacco. Now the genetically modified tobacco glows after spraying with luciferin, the substrate for luciferase and ATP molecule. And later on, of course, green fluorescent protein from jellyfish is also used to develop glowing plant. So these, growing plants may have wonderful applications in the coming future. For example, if one wish to extend this technology, bioluminescent technology, you can make bioluminescent landscapes or people's yards or bioluminescent grass or even growing Christmas tree with a naturally, showing glow. You can also produce trees, growing trees, which can be planted on either side of the road or streets. So I'm not saying that glowing plants can replace electricity, but they can be used for some of these important applications in the future. And recently, a very potential technology was discovered. It's called RNA interference or interfering RNA. This technology is known as a very potential gene silencing technology, and this has many applications. These are the two guys, Andrew Foyle and Craig Mello from US. They have discovered RNA interference phenomena for silencing the genes. So basically, the double-strand RNA, whose sequence is complementary to the target messenger RNA, can be used for gene silencing. So now we know that RNA interference is a highly conserved mechanism across eukaryotic systems, and RNA is also involved in innate immunity against viral infection. So the RNAi potential gene silence technology can be used for a variety of potential applications in agriculture. Like I've already covered potential applications of transient technology. All those applications, all the implement of all those economically important characters virtually can be done using RNAi. But the difference is here, this is a gene silence technology, the conventional transient technology, where mostly we overexpress a foreign gene. But here, we downregulate a target gene either in the plant or the target pathogen or insect for engineering useful traits in croplands. The RNA interference silence technology is also used for basic application, that is in functional genomics. In other words, you can decipher the function of newly isolated genes using RNA, because RNAi can create a very good mutant phenotype through silencing. So I'll give you very few examples of RNAi application in agriculture. RNA interference technology is very successfully used for development of virus resistance, nematode resistance, insect resistance, as well as fungal resistance. How these were developed, they have identified a vital gene in the pathogen like virus or pests like nematode or insect pests, and then they could knock down the expression of those identified target genes by expressing double-strand RNA specific to those target genes. So this technology is also called host-induced gene silencing, because you are silencing a pathogen insect gene through host plant-mediated RNAi mechanism. In fact, I have been working in this area since last about 15 years, particularly for the development of virus and fungal resistance, and also insect resistance. Dr. Abhinav has already mentioned in the beginning. So RNA technology can also be used for a variety of other applications, like one can develop abiotic stress-tolerant crops. Blue roses have been produced, gossipal-free cotton, even slow ripening tomatoes. You name anything which can be done by RNAi. People could even develop decaffeinated coffee, nicotine-free tobacco, or high lysine rice, etc. There are many exciting applications of RNA technology, like the use of transgenic technology. And very recently, there is an excitement about genome engineering technology or gene editing technology. And here, so this technology is very more precise, and this technology can be used either for gene deletion or for creating a mutation, insertion, or at a particular site in the DNA. Basically, there are three different technologies which are basically the endonucleases. They can be used for sequence-specific, gene modification. But among these, like zinc finger nuclease or tall effector nuclease and CRISPR-Cas system, among these three, CRISPR-Cas system is more preferred because of its specificity. And this technology is very successfully used for inserting a mutation or removal of a gene or knockouts. So in conclusion, the plant tissue culture technology and transgenic, including RNA interference, and also the microRNA, artificial microRNA technology, which I have not discussed because of time limitation. So they have a great potential applications in agriculture for crop protection against pathogens and insect pests, as well as the improvement of many other agronomical important characters. So these fields, particularly RNAi, this has opened up a new functional genomics of crop improvement and food security. Finally, I will take a couple of minutes more to discuss about global area of biotech crops. And the first biotech was commercialized in 1996. So since last about 18 or 20 years. So now the more than 400 million hectares of land is being cultivated by biotech crops. That too, a very few crops like BT cotton, herbicide-tolerant cotton, herbicide-tolerant soybean, and BT corn. Mostly, these few crops are cultivated in more than 28 countries, and so this has led to a very, it's very beneficial to the farmers economically, and yeah. Although more than 20 important crops have been commercialized globally, there is a great concern about the cultivation of genetically modified crops and also consumption of genetically modified food by some people, particularly people from Europe. Where the most of the European countries they don't accept the cultivation of biotech crops. There are even many myths about genetically modified foods. I'm not discussing all these, but I would like to mention this particular point. Well, somebody has, for example, somebody has introduced a gene from fish for omega-3 in strawberry. You can see the cartoon. The strawberry is looking like fish because it has fish gene for omega-3. In India, as you know, more than 50% of the population are vegetarians. There's a myth that they might think that strawberry is no more a vegetarian food. Now, it is non-vegetarian because it contains a gene from fish. This is one of the myths, and there are many other myths, like some people even think that genetic engineering is illegitimate recombination, right? Many other. But the true reality or the truth is the genetic modification is happening since ages. As I would like to give this particular example of corn. On your left panel, that is a wild species of corn, which is called teosinte. You can't even make it as a corn. And it took about, you can't even see the kernels or seeds. It took about 5,000 years to come to this stage, the middle one, where you can see the kernels, and it took another 5,000 years for the evolution of modern corn. So it took totally, it took about 10,000 years for the evolution of modern corn. How this has happened? This has happened due to the mutations or mutation and recombination in our interspecific hybridization. So through genetic modification, the modern corn is evolved, and this is very true with many other plants, right? So mutation, genetic modification is happening in every organism since ages. So why people are concerned about genetically modified plants or organisms, where the gene of interest with known function is introduced, right? So, but for example, radiation breeding. in the classical breeding, people also use radiation for creation of mutation or for widening genetic variation. Some people think that radiation breeding more dangerous because radiation can induce a variety of mutations, including chromosome breakage randomly. So some think that this is more dangerous than than the other techniques of genetic engineering. Look at this slide. Actually, this girl is eating popcorn from genetically modified corn. Initially, she doesn't know. When she was told that you are eating popcorn made out of genetically modified corn, then she started screaming that you can't listen to the screaming. Then I would like to have your reaction, how you react yourself if you are provided with a genetically modified food. I'm sure being students, including genetics background, I'm very sure you would accept genetically modified food. Currently I have two major research programs involving transgenic approach as well as gene editing technology, CRISPR-Cas system for engineering virus resistance in papaya. And in case of rice, of course, we are trying to improve the yields by transgenic technologies, including overexpression of microRNA genes. I would like to thank my research team for their active participation in my research programs, and also the funding agencies, particularly Department of Biotechnology, Department of Science and Technology, for generous funding my research programs. Of course, I have not discussed it my own work because I was told that most of the participants have BTech background. Some even have 12th standard or PG. I hope you enjoyed my talk. Thank you very much. And if you have any questions or queries, I would be pleased to answer. Yeah.
Yes, students, if you have any questions, you can ask.
Jyotika Sahu.
Okay.
Roma, you have to unmute yourself. You are muted.
Yes,
Should I ask? Yeah.
Jyotika, you can ask.
Jyotika?
What is the medical use of spider silk?
Pardon?
Hello?
Hello, yeah.
Sir, I was asking what is the medical use of spider silk?
Your voice is breaking actually.
Can you
repeat? Yeah.
Sir, what is the medical use of
Jyotika, your voice is not clear, so you can write your question in the chat. I'll just.
Jyotika, are you asking that what is the use of a spider silk?
Yes.
Medical, she was mentioning some medical use.
Yeah, medical use of a spider silk or what?
Medical use of spider silk. Yes, I was asking.
You just type your question.
In the chat section, you can type your question.
And the chat is disabled.
No, I'm sorry, it was not clear. There's an internet problem, I think.
Exactly.
Any other question?
Anyone wants to ask any question? I'll just unmute.
Yeah.
Any other student?
Yeah, sees Ankit Verma is asking, so what is the medical use of a spider silk?
Okay. Like as I mentioned, spider silk protein is being used for the preparation of, the sutures, in operations, surgical operations. But there are many actually. Yeah, this is one of the important applications of spider silk protein. Yeah. Any other questions?
I think there are no more questions, sir. One more question is there from Harsh Kumar Sahu.
Okay.
He is asking how bioluminescent plants are developed.
Yeah. For developing bioluminescent plants, you have to introduce lux gene from insect fly or lux gene from bioluminescent bacterium. But unfortunately, the plants do not have the pathway leading to luciferin, which is substrate for luciferase enzyme. So you may have to introduce a couple of more genes to have Lucifer synthesis in plants along with the Lux gene so that the plants can eliminate light. They show glow or bioluminescence. Yeah. So this is through gene introduction. And more recently, people are also using green fluorescent protein, which was isolated from jellyfish for development of, glowing plants. And I've already mentioned various applications of bioluminescent plants, but these are future possibilities. So far, bioluminescent plants are not used for these applications in nature.
Sir, one of the students is asking, how is biosteel used in medical field?
Yeah, but the biosteel, that is spider silk protein, is most commonly used for sutures, in stitching during operations. Yeah, but there are a few other applications.
One student is asking, sir, are there any side effects of using food that is made up by help of biotechnology? I think he wants to
Yeah.
ask genetically
Okay.
modified foods.
Yeah. See, till now there is no strong scientific evidence of negative effects of eating genetically modified food. So, for example, the flavor saver, slow ripening tomatoes were commercialized in US about 15 years back, and American they ate tomatoes, genetically modified tomatoes for many years. There's not even a single case of negative effects of eating genetically modified tomato. Likewise, I mean, because these genetically modified plants, they are only commercialized after thoroughly testing them against allergicity, toxicity, and also aggressiveness towards environment. So usually it takes about 6 to 7 years for rigorous testing of genetically modified plants before they are commercialized. So they have to, the genetically modified food as well as plants, they had to be substantially equivalent to their counterpart, the genetically modified plant or wild type. Then only they will be commercialized, right? It's like a new medicine. If somebody discovers a new medicine, the new medicine has to undergo many clinical trials. Usually it takes about 10 to 14 years to develop a new medicine. Similarly, the genetically modified plants, usually takes 7 to 8 years for development as well as testing them against the toxicity, allergicity before they are commercialized. So far, to the best of my knowledge, there are no side effects of eating genetically modified food because they are thoroughly tested on several model animals, including monkeys, sheep, etc. Yeah.
Sir, one more question is there. A student is asking how Bacillus thuringiensis strain can be used to eradicate the pathogens in cotton and brinjal.
Okay. Say the Bacillus thuringiensis, it has a protein called delta endotoxin. The delta endotoxin is highly insecticidal, right? It can kill most of the insects, particularly the insects which belong to Lepidopterae, like Helicoverpa armigera, which is called as cotton bollworm. And so, in fact, since last more than 40 years, the Bacillus thuringiensis, the bacterial suspension, is being used as a spray for the control of a variety of insect pests. And then later on, the cry genes, which are responsible for endotoxin protein, were isolated from different strains of Bacillus thuringiensis, and they are being transferred not only into cotton but also other crops like corn for engineering insect resistance. Yeah, and these are highly effective against Lepidoptera insects, and also insects belong to other groups like Coleoptera and all. Yeah.
We will take one last question. So
Yeah,
there's a
sure.
question from a project fellow, Alok Dubey. He is asking, is there a way to deal with cry resistivity?
Yeah, I briefly mentioned actually. So initially, the Monsanto company and some other companies, they have introduced only one gene, that is Cry1AC, for resistance against Helicoverpa. But after some years, the Helicoverpa has gained resistance against Bt protein. So therefore, they had to introduce a second gene, Cry1AB or some other gene, to take care of the insect resistance managed. So these are called Bolgut2. They have Cry1AC as well as one more gene. So if insects, the target insect, if it gains resistance against one protein, but the crop is still protected because of the presence of second gene. So this is how one can manage insect resistance problem, but there are few other strategies also. One of those strategies is a refugia, where where you can have 20% non-GM around the GM crop, right? The insect attack the non-GM first, right? So that it won't develop resistance, against BT protein. Or alternately, one can express BT protein in huge quantity, so that the insect is instantly killed. So there are, these are the different strategies for the management of insect resistance problem. Yeah.
Okay, students, we will not be taking any more questions due to shortage of time.
Okay.
Personally, we will be taking up the modules on plant technology. So you can ask Dr. Abhinav your remaining questions when he takes the module. I would like to express my heartiest gratitude to Rajim sir for taking out his time and interacting with us. It was a very insightful session, sir. Thank you. And we got to know about a lot of applications from plant tech and the future of plant technology. So thank you very much, sir.
Thank you, Abhinav, and thank you.
Thank
Yeah.
you.
Thank you.
Thank you so
Bye.
much, sir.
Yeah, thank you. Bye.
Bye, sir. I request the students to please stay in the session. I have to talk. Sir, you can stop sharing the screen.
Yeah, just I'm looking for.
I think sir को वो leave meeting वाला option पता नहीं. I request all the participants to switch on their video. We have to take some photographs of the inaugural session. So video has to be on. All the videos have to be on. Gitanshu, Samantha, Priya, Nandini, Shruti, Mayank. Please switch on, turn on your videos. गीतांशु, हर्ष कुमार, आपका वीडियो ऑन नहीं है. गीतांशु, आपका वीडियो ऑन नहीं है.
I guess there is some connectivity issue.
शिवानी,
हर्ष कुमार साहू.
वो मैं फर्स्ट स्क्रीन का ले रही हूँ, बाकी का तुम ले लेना, ठीक है? Okay, so I want to know from all the participants the feedback of the first day sessions. How did you find the lecture of Pooja ma'am and Rajam sir? Was it helpful for you all? I've unmuted everybody. Feedback.
It is helpful for.
It was very helpful.
हाँ सर, नो पीछे से ये ना जब भी कोई सेशन अटेंड करते हैं तो गाने वाने नहीं बजाने चाहिए, नहीं बजने चाहिए प्लीज. I have not yet received the undertaking from remaining students. बच्चे जो हैं बच्चे ही रहेंगे.
See, there are two options. When we are muting you, you can just raise your hand. या जब हम आपको अनम्यूट करते तो आप खुद से म्यूट कर लिया करिए अपने आप को, ठीक है?
जिसको बोलना होता है वो, हाँ, जिसको बोलना होता है वो खुद से अनम्यूट कर सकता है. हमने म्यूट किया होता है. You have the option to unmute yourself. So when you want to ask any question, you should unmute and then ask. ठीक है? And attendance we will be taking in 2 slots every day at 10 o'clock and at 1 o'clock. So you have to be there from starting till the end. And we'll be conducting the quizzes by Google Forms. I had shared this in the mail also, that in the second half, a quiz will be floated to you all. You have to fill it by 9 o'clock in the night. 9 p.m. is the deadline for that. And your assessment, I'm going to tell, we are giving 40% weightage to the quiz and 60% weightage to the exam, which will be conducted on 5th of June, 2-hour exams. So every Friday, the quiz that is conducted on Friday will be a graded one. That will be counted in your final assessment. So you will get practice quizzes from us every day, but Friday quiz is going to be counted in the grading. And all the participants who have more than 80% attendance, who score more than 50% in total, including quiz as well as external, they will be getting the certificate. So make sure to give your original name and your affiliation, क्योंकि certificate पे वही print होगा जो नाम आपने दिया है. So I request you to take one, take this from them, the data, that their original name and affiliation should be there for
Yes,
correct
yes.
preparation certificates. Right?
Your name and details regarding your organization must be the same, what you have registered. If there is any kind of modification, then I'll circulate one more Google form. Okay.
So tomorrow we resume with the session at 10 o'clock. I will share the ID, Zoom ID in the evening for tomorrow's session. So tomorrow we will have 3 lectures, one from me on introduction of biotechnology, industrial biotechnology, one lecture from Roma ma'am on introduction to biomolecules, and one lecture from Abhinav sir on media. So see you tomorrow at 10 o'clock.
Thank you so much, ma'am.
Okay, ma'am. Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am.
Thank you so much, ma'am.
Thank you, ma'am.
Okay, take
Thank
care.
you, ma'am.
Take care.
Keep seeing your WhatsApp group. You'll get the link for tomorrow's ID on that only.
Thank you so much.
Okay, ma'am.
Okay, ma'am.
Okay, ma'am. Thank you, ma'am.
Okay, bye-bye.
Okay, ma'am. Thank you.
Okay, Pallavi ma'am.